amfAR HIV Cure Research Institute, and I want to start by thanking amfAR. Well, I'll introduce Kevin Frost in a few minutes, but thank them for putting their support, their trust in us. We think we have a great team. You're going to hear about our team today, and the relationship that we have with amfAR is a really important one. So we welcome them here, and we welcome this opportunity. Before I forget, I'd like to thank a couple staff members, Jessica Sapphire and Becky Cantor, who have worked really tirelessly on the Cure Institute and on this program, and also to all the amfAR staff that are here as well working on this. I'd like to thank our entire San Francisco community, and especially the people living with HIV that are here with us today. AIDS Day is always a great opportunity to remind ourselves how fortunate we are to have done what we've done and survived this plague, but to also remember the people that we've lost along the way. Too many, obviously, to count. And because we want to move as quickly as possible towards a cure in the amfAR Institute, our involvement with the community is especially important because when Steve Deeks talks about the clinical trials that we're going to be doing, it's obviously the community volunteers as participants that are really important, essential to make that go as quickly as possible. I'll give you a quick overview of our team that we have assembled for this institute and of our scientific approach to this. We're going to make the point several times that this field is still evolving so quickly that we want to be prepared to pivot if something changes and looks more interesting. But so far, we're happy with our initial decision, and I'll talk about that, and you'll hear more about it later. I'll try to keep us moving along quickly so we have time at the end. We'll collect all the questions that you have. There are question cards at your seats. We know many of you are very involved in this issue. We look forward to the questions, and we'll do a panel discussion at the end of the program before we invite everyone to stay around for a reception outside afterwards. So let me start with my slides. This is our mission. I'll read it. The mission of the amfAR Institute for HIV Cure Research is to harness the innate and adaptive immune response to reverse latency, reduce the reservoir, and you're going to be hearing a lot about the reservoir, and control what's left. This is the model of the amfAR Cure Research Institute. We felt that the problem of curing HIV disease is too big. It's still a very seriously complex problem, and that we thought it would just take too long with a conventional academic model. And so with amfAR's involvement, we wanted something that would allow us to move as quickly as possible in a real solid direction. So the amfAR Institute contains many scientists. We have several industry partners. I'll talk about them as well. There are multiple scientific disciplines involved in our team, and we are assembled into effective teams. I think most of us have never worked quite as closely as a team until this project came along. amfAR really wanted this to be a team at a place, and so even though we have other research projects as individuals, we came together as a team for this work. And we know that unless we set milestones, unless we measure what we're doing, we won't know that we're making progress. We want to make progress. We want to convince amfAR that we're making progress, and we want to convince the community that we're making progress. So we're setting milestones to hold our own feet to the fire. Here are some of our partners and affiliates. Obviously, the University of California, San Francisco, the Gladstone Institutes, and the Blood Systems Research Institute. Both of the second of those are close affiliates, academic affiliates of the university, but we also act independently. but we're working closely together in this program. We also work closely with the Oregon Health and Science University in Portland, and we have collaborators working with us from UC Berkeley as well. Our two primary industry partners are IDRI, the Infectious Disease Research Institute. It's a nonprofit company based in Seattle that, and we'll talk much more about this, that makes products that help vaccines work more effectively. And Gilead Sciences, based here in the Bay Area, which is a very exciting research program in HIV cure that's now part of our group. We talk in our group a lot about definitions of what is a cure. And I know a number of you have thought about that and participated in some of those discussions yourselves. But I wanted to start with my kind of non-definition summary of what I think we want to achieve. We want to allow an HIV-infected person to live a full life without having to take medicines, without having to worry about transmitting the virus, and without having to suffer any complications from HIV infection. So if we could do that, we would have done a lot, and that's our goal. There are different definitions, and I think some of the speakers, I know Steve Deeks will talk about this as well. People talk about, well, is curing it like Timothy Ray Brown, where the virus seems to be completely gone from his body? And that would be great. That would be a true cure, or what we might call a sterilizing cure, where every HIV-infected cell in the body has been eliminated. No one would question that that's a cure. But along the way to that, we talk about a functional cure, which is more like a remission of a cancer, where it might come back, but where for at least, we would hope, prolonged periods of time, people could live without taking daily HIV medicines. Perhaps we would do something to reduce the size of the reservoir, and then do something to maintain the body's immune control of what's left of the reservoir as, again, a step towards the final cure, which would be an eradication. My slides are not, there you go. So when amfAR put the Cure Institute together, when they put the proposal together to which we responded, they said, let's think of the four main questions that might help define the areas of science that would be involved in this. And they came up with an acronym, CURE. Not surprising, perhaps, but what this stands for is charting the location of the reservoirs, understanding how reservoirs are established and persist, recording the amount of virus in reservoirs, and then finally eliminating the reservoirs. And we've divided our science largely around these areas of work. And when I talk about the main questions that our scientists are addressing, it all fits into this acronym. Oop. We have to look at our photos. So this is our team, and it's expanded from last year when we first introduced the concept of this institute here in San Francisco. Really, I think exactly a year ago today. The board of directors, we call ourselves, are the top row plus Rowena Johnston. You'll meet each of the people on the top row in the presentations today. Rowena couldn't be with us, but she's the scientific officer at amfAR, and is a very unusual arrangement. We have the funder really sitting at the table with us when we talk about our science. So in all of our regular meetings, Rowena is part of that. It's a unique mechanism, and we hope that it's one that's going to allow us, again, to make more rapid progress as we're integrated more fully with the rest of the amfAR Cure Research portfolio. On the bottom are other key members of the team. Afama Yoke is a scientist in Oregon. Terry Liegler and Jeff Malouche run our basic science core. And Linda D., we're happy to announce, has agreed to be the co-chair of our community advisory board. We're in the process still of forming that, but she has volunteered to help with that, and we imagine a community advisory board that's based locally and nationally, because this is the national amfAR Cure Research Institute. Now, how did we get started? We talked about this a little bit last year, but there was one experiment that we saw. It's actually still not published, I think, where scientists at Gilead had a substance. It's called a toll-like receptor, TLR, and I think that's the last time you'll see toll-like receptor spelled out. So when we say TLR, that's it. And what it is a stimulator of the immune system. And there was evidence in this trial in monkeys that this TLR7 that was obtained from Gilead Sciences was able to activate cells in the latent reservoir, causing them to produce virus, which is, you'd think that's not a good thing, but it is a good thing if you want to stimulate the reservoir as part of a therapy to cure the infection. And what we saw was that this compound was able to cause blips, periods of viremia after it was given, in an otherwise completely suppressed HIV-like system. And so this was very interesting to us. It suggested that this compound, which can be given orally, might be the kind of shocking agent that we've been looking for in this overall shock and kill type approach. So this was one of our first decisions, was to go with TLRs as our shocking agents. There's another study, though, that gave us a lot of pause and still does. This is a study, the first author, Tim Henrich, is now here with us in San Francisco, working in the Division of Experimental Medicine at the Zuckerberg San Francisco General. And what this study showed was that in patients, these are the Boston patients, people that had a bone marrow transplant and appeared to be controlling their HIV afterwards, but then both of them broke through. But they broke through at a very long interval after their procedure without antiretroviral therapy. We think that the procedure decreased their reservoir perhaps to as little as one infected cell, and yet they broke through. And so what this slide teaches us, what this data teaches us, is that our shocking approach would have to be really, really effective in order to hope to eliminate the virus from the system. So it suggests to us that we don't have anything that can do that yet, that the first approach of shocking will have to be combined with something else, and you're going to be hearing more about that from the rest of the program today. What recently gave us a sense of encouragement, though, was this study. This is another monkey study. It's a study in which monkeys infected with a form of HIV were taken off antiretroviral therapy. What you see is that each of the groups rebounded when the therapy was discontinued, but compared to the groups that had nothing done to them, or ones that had just the TLR7 again, or TLR7 plus a novel vaccine, they rebounded and stayed pretty viremic. But in contrast, the animals that got the combination of the vaccine and the TLR7 seemed to do much better. They seem to be much more able to control their viremia. And this then is very exciting because we are in the process of developing a direction in our institute where we will combine TLR agents with vaccines to hopefully achieve this kind of additional control. We think that the vaccine then is, a therapeutic vaccine is helping the immune system to suppress the ongoing viremia. So what about the scientific programs? I won't orient this along those modules of C, U, R, and E, but each of the questions that I'm going to pose is led by one, two, or more of the scientists that you've been introduced to, and I should say supplemented by the work of a number of other scientists, some of whom are in the room today. So it's not as though each of these people are doing all of the work themselves. They have an entire team of people in their laboratory. So Warner Greene will ask questions about the biology of TLR. Questions that his group will be addressing are, how effective are these drugs in reversing latency in the HIV reservoir? We've seen suggestions from the monkey study that they might activate the virus. We're going to look for that in the laboratory. Can they be used in combinations? There's some question about whether TLRs might be too toxic to use by one at a time in full doses. So maybe by combining them, we can find combinations that allow us to decrease side effects and increase effect. Peter Hunt and Satish Pillai are also very interested in TLR biology, and they're going to be measuring this and asking the questions, can the impact of TLR stimulators, the drugs that we're going to be using, in reducing the HIV reservoirs in tissues as opposed to circulating blood, be measured? Satish will talk about this and talk with a nice model that he's come up with about how do we think about the circulating virus versus the virus in the tissues, which we think is much more important. Do the cellular reservoirs that stay in the tissues respond differently to TLR stimulators than the reservoir cells in the blood? And maybe by looking in the blood, we're seeing the wrong impression compared to what might be happening in the tissues. So this is going to be a very important part of our work. Can highly sensitive tests be used to measure the dangerous form of HIV that can grow actively and spread to other cells? We talk about replication-competent virus. This is a virus that is fully armed and ready to grow and infect new cells. How do we measure that most accurately? And some very interesting work that's starting is, can the effect of TLR stimulators on these tissue reservoirs be actually visualized using radio labels and combining that with imaging that sort of is related to the MRI that most people are familiar with. So actually trying to see this virus radiologically. Steve Deeks and his colleagues, including Ashley Haas and Tim Shacker, are interested in asking, can we show that any of the treatments that we're using and the reservoir measurements that our team is going to be developing predict how long HIV will take to relapse once HIV medications are stopped? This is a really central question. We think that the lower the reservoir, the longer it takes to relapse, but that's the kind of question that we're going to be asking. And to do that, we're actually going to be starting a clinical trial very shortly. And again, we are going to be looking for people who volunteer for this important work. This is going to be a trial that we call an analytic treatment interruption, where someone on stable antiretroviral therapy has the treatment stopped. We observe very, very carefully. We know that the virus will come back, but we're interested in how long it's going to take to come back and whether markers that we develop can actually predict how long that process will take. That would give us some clues into how we would conduct further trials when we have more effective research strategies. Again, in collaboration with our scientists at Oregon, Louis Picker and AFAM, Steve Reed from the Infectious Disease Research Institute, and Romas from Gilead, we're going to be asking whether we can measure TLR stimulator activity in primates and humans. So this is when we get to the issue of, when we have better directions, can we move into human trials and actually begin cure-related programs? Should various TLR stimulators be used alone? As I said earlier, there's some thought that maybe combinations would be more effective or less toxic. How are these drugs best administered? Can they be given orally, as in the case of the Gilead drug? Do they have to be given by injection? If so, do we give that by a subcutaneous injection under the skin or IV? So these are the questions that we can address using the primate models first before we bring these compounds into human trials. Now, we have a program that you're going to be hearing about today that we think is exciting. And one of the things that's most exciting is that we have the occasion in San Francisco of having some amazing investigators on the team who are also doing work funded by other groups. The DARE collaboratory that Steve Deeks directs, that Steve and Mike McCune, who's in the room, directed until recently, are going to be working in parallel with the amfAR Institute. We're going to be making sure that we don't duplicate efforts and that we hopefully don't leave any great gaps in our research approach by looking at the various ways that we might approach HIV cure. We want to reverse immune suppression, wake up the immune system. We want to lower the reservoir by these shocking approaches that we've been talking about. We want to learn how to measure these effects very much better than we've been able to in the past, and we think that we have the team that will be able, with this support, to make progress in this area. We are proud to be here at UCSF and the Mission Bay campus. This is our this building across the kind of diagonally from this building is the Mission Hall. It's where the amfAR Cure Research Institute is based, and we're now getting signs put up in our garden. So the garden area behind the building is going to be the amfAR HIV Cure Research space as well. So we are really happy to be here today. Again, thank you for coming to the program. We really do look forward to participation and questions that you might have as the day goes on. And I think with that, I'll introduce Kevin Frost. Kevin is the CEO of amfAR and a long-standing colleague. Kevin has been involved in HIV work for decades, like some of the rest of us at this point. And we look forward to you. And Kevin will then introduce the rest of the speakers. Thank you very much.
Thanks, Paul. Thank you. Well, good afternoon, everyone. It's nice to see everybody here again at our second annual summit on HIV Cure Research here at UC San Francisco and at the amfAR Institute for HIV Cure Research. Let me add my welcome to Paul's and also my thanks to the UCSF staff, but also to the amfAR team members who are here for all the work that they did to bring this program together. I do want to recognize one person who's in the room who probably needs no introduction, but he is an amfAR trustee since day one of the epidemic, and you all know him, I'm sure, here in San Francisco. Dr. Merv Silverman is here. Thanks, Merv, for being here. So I'm excited about this summit for a couple of reasons. Today I get to play moderator, and so what we're going to do is we're going to have a series of presentations, as Paul said, and when you sat down, I'm sure you all saw the cards that were at your seats. I want to encourage you to write down your questions as we go along and pass those up, and then at the end of the program, we're going to get all the presenters up and we're going to have a little discussion amongst ourselves, and we'll go through the questions at that time and try to get them all answered. But I'm also excited because the summit does two things for us. First of all, it gives all of us, myself included, an opportunity to get updated on the science. And this is complicated science. And it's hard to understand when the last science course you ever took, in my case, was a freshman biology course 30 years ago. It can be a little daunting. But the other reason that I'm really excited about it is because you get an opportunity to hold our feet to the fire. Because we've set out, as Paul said, some real milestones in this grant. And we want to understand how we're doing. And we'd like all of you to let us know how we're doing. And you can do that any of a number of ways. You can talk to us at the end of the program. You can email me. I'd love to hear from you. My email is really simple. It's kevin.frost@amfar.org. If you have anything you want to share with us about this program, how well you think it's going, not going, this is the chance to really help us make sure that we're on track to get where we want to be. So please, speak up. So as we get going, I said a minute ago the science is complicated, and it is. And at amfAR, we've struggled over the last couple of years as we've gotten deeper and deeper into this cure research field to find ways to talk about it. We've struggled to figure out how to explain the work that's going on and what we're trying to achieve. Paul talked about the shock and kill approach, which is going on here, but there's a tremendous amount of other research that's also going on in areas of gene therapy, for example. And the Institute forms the cornerstone of our cure research efforts. But we've made a commitment to invest $100 million over the next 5 years. The Institute represents about 20 million of that, but in the first year and a half alone, we've already committed $40 million to this effort, and in the next several years, we're going to commit that full $100 million in this research. But how do you explain to people these complicated concepts around cure research? And the one word that keeps coming back, the word that you're going to hear over and over and over again, is the word reservoir. And I think that the reason the reservoir is so important is because I do believe there's broad consensus in the scientific community that the barrier to achieving a cure is overcoming the reservoirs. How do we deal with these reservoirs? So what we did over the last many months, and hard work on the part of our team at amfAR to do this, was to create a series of animated films that we hope will help people to better understand what these reservoirs are and what the approach is that we're going to take or that needs to be taken in order to address them. So by being here today, you're going to be seeing the world premiere, the first public showing of each of these animated films. There are 5 altogether. I'm going to start by showing you the first one now because it gives you an overview of the whole reservoir concept and why that's important. And then after that, I'm going to talk about, as we introduce each of the 4 speakers, we're going to talk about how each of the modules that form that CURE roadmap, the C-U-R-E roadmap, how each of those play into the science and how it's represented in their work. So we're going to begin now by showing the first video to give you an overall sense of the reservoirs and how they operate. So if we could show that first one, thank you.
When HIV, the virus that causes AIDS, enters the body, it zeroes in on CD4 T cells, its preferred cellular target. Once inside the cell, the virus makes its way to the nucleus and inserts itself into the cell's DNA. Most infected cells then begin to actively produce new virus. HIV virus particles, or virions, bud off the surface of the infected cell and go on to infect new cells. But a small number of infected cells do not actively produce virus. Instead, the virus inside them is inactive, or latent. During this latent infection, virus remains in the DNA, but does not produce new virions. Our immune system normally protects us from viruses. For example, specialized immune cells detect surface proteins that signal infection and kill the infected cells. However, infected cells that contain latent virus do not produce such surface proteins and therefore are not flagged by the immune system. The virus in these cells lies dormant, waiting. These cells are indistinguishable from healthy cells and are known collectively as the viral reservoir. It's important to understand that this reservoir is not a place. It's a collection of sleeper cells in major pockets throughout the body. When a person starts taking ART, any virus that is produced cannot infect new cells. This is why a person must take antiretroviral therapy their entire life. Otherwise, any virus that is produced can spread to neighboring cells and beyond. To remove the need for ART, we need a cure. To cure HIV, we have to eliminate the reservoir that is the source of active infection once a person is off treatment. Timothy Ray Brown, the Berlin patient, gave the world proof that removing the reservoir is possible. After a stem cell transplant of naturally HIV-resistant immune cells, the virus could no longer be detected in his body. Scientists and doctors concluded that removing the reservoir led to his cure. In our efforts to cure HIV by eradicating the reservoir, we have learned a great deal about how to achieve a state of viral control that we call remission. By studying the small number of people who are able to control their virus without ART, we may be able to replicate this state of remission in others. To accomplish cure or remission, we need to chart the locations of the viral reservoir, understand how HIV persists in this latent state, record the extent and size of the reservoir, and figure out how to disrupt or eliminate it. This is our roadmap to a cure. It would transform millions of lives and would pave the way to ending the global AIDS epidemic in our lifetime. Join the countdown to a cure for AIDS.
All right, so I hope that gives you at least some idea of what we're talking about and a lot about what you're going to be hearing. I'm going to introduce the next video right away because I'm not going to spend any time introducing the speakers. Our first speaker is Peter Hunt, and if you want to know who Peter is, look in your programs. He's an associate professor here, but you can look in your programs and learn everything you need to know about Peter. But more importantly, this next video is going to talk about charting the location of the reservoirs. It's about figuring out what cells the virus resides in and where they live. And so part of understanding the roadmap is figuring out this very, very difficult question. We're going to show the video, and I'm going to turn it right over to Peter.
The main obstacle to an HIV cure is the reservoir of virus that is able to stay under the radar, evading attack by the immune system and powerful anti-HIV drugs. Pinpointing, or charting, the location of the persistent HIV reservoir is key to ensuring that a curative intervention targets the correct cells. Although the infected cells serving as a reservoir of HIV could be anywhere, they tend to congregate in certain hotspots in the body. We know that certain tissues, such as the gut and lymph nodes, are major pockets of reservoir, but other sites are starting to be studied. Beyond the anatomical locations of the reservoir, we also know that the reservoir prefers certain types of immune cells, especially CD4 T cells, over others. Like an extended family, CD4 T cells are made up of many related but distinct members with different anatomical locations, ability to activate, and prevalence. Latent virus can exist in all of these in different proportions. It's important to know which types of cells contain the reservoir so that specialized interventions can be developed to more effectively target those cells. Within each reservoir cell, even the location of the integrated virus, the provirus, inside our own DNA, needs to be charted. In most cases, the virus inserts itself in a way that results in active virus production, but the virus may become latent if it is inserted in a way that decreases or even prevents its own activity. The virus may, though, become active at a later time. Knowing the precise location of the reservoir anatomically, by cell type, and within the DNA itself ensures that our cure approaches target the reservoir in the most effective manner possible. Join the countdown to a cure for AIDS.
Okay. So thanks, Kevin, for that video and the introduction. I'm going to talk about the work that we've been doing in the charting module of the amfAR Institute. And we're particularly interested in charting the reservoir of HIV in tissues. This is our lead team of investigators, Mike McCune, Shomi Sanjabi, Nadia Rohn, Zoltan Lasik, and Steve Uckel. And so why are we focused on the HIV reservoir in tissues? Well, CD4 T cells that you just heard about in the video, the major reservoir of HIV, traffic through the blood, but they really live in the tissues. In fact, only about 1% to 2% of the total body's CD4 T cells are in the blood at any given time. The vast majority of them reside in the tissues, like the lymph nodes, the spleen, and the lining of the gut that you heard a little bit about in the video. And in some ways, if you think about it, these lymphoid tissues are sort of like cities where most people live. Most people are concentrated in the cities, not in the rural areas. And blood is perhaps like the highways that connect cities. People travel between cities on the highway, but at any given time, far fewer people are on the highway than are in the cities. So it's not just that fewer people are in the peripheral blood or fewer HIV-infected T cells are in the peripheral blood, but those cells may be very different than the cells that lie in the tissues, as Kevin was alluding to. And we get a hint of this by thinking about whether people in the cities are different than people who live outside of the cities. I think the recent election a couple weeks ago gave us a strong sense of that, right? People voted very differently in the cities than they did outside of the cities. And the same is very true, we think very much true, for infected T cells in the body. HIV-infected T cells in the tissues may be different than those that we find in the blood. First, the proportion of CD4 T cells that are actually infected by HIV is at least 10 times higher in the gut than it is in the peripheral blood. So far more of those cells not only are present there, but a higher proportion of them are infected in the gut than in the blood. So that's different. Some types of infected CD4 T cells in the lymph nodes in the gut don't even circulate through the blood. So if we're only getting peripheral blood draws, like in the doctor's office, we're never going to see those specialized subsets of cells that live in the tissues. Many of these cells may also respond differently to therapies designed to either shock HIV out of its hiding places, so shock cells that are latently infected to start producing virus again, and also therapies to help kill those infected cells once they're producing the virus. And for these reasons, we really need to be able to measure the virus in these specialized CD4 T cells in the tissues. So let me tell you about some of the work that we're doing here. The first team of investigators, led by Shomi Sanjabi and Steve Uckel, has been focusing on a specific type of cell that you saw briefly mentioned in the video called T follicular helper cells. Let me tell you a bit about these cells. These T follicular helper cells, or TFH cells, are a major reservoir of HIV in lymph nodes and in certain locations in the gut, like the terminal ileum. These TFH cells live in the B cell follicles of the lymph nodes, and they normally provide help to B cells to make antibodies, a key part of the immune system's function. But it turns out that these cells, they usually stay in the follicles and they don't circulate in the blood. So this is another one of those cell types that if we're getting a blood draw, we're not going to find them. And these TFH cells are primary targets of HIV in the lymph nodes. HIV preferentially infects them, and these cells produce the most virus in the lymph nodes in untreated infection. And that's what you can see in the slide on the right here, the F here standing for follicle. This is the B cell follicle, and all the pink stain is virus production. You'll see it's not randomly distributed throughout the lymph node. It's concentrated in these follicles where the T follicular helper cells are producing tons and tons of virus. But it's not just that these cells are preferentially infected and producing virus in the lymph nodes, but these cells may also be protected from killing by the immune system. So the B cell follicles where these TFH cells live is a so-called sanctuary site for the immune system. So many of the cells that would normally kill HIV-infected cells are prevented from entering the follicle. There's like a brick wall protecting these purple TFH cells that may be infected with HIV in the center from being cleared by all these killer cells in green on the outside of that brick wall. For this reason, these infected TFH cells may be more difficult to clear with immune-based interventions designed to rev up and increase the function of these killer cells out here. Therapies like the TLR stimulators that Paul spoke about just a few moments ago. And it may be, if that's the case, that we need additional interventions that poke holes in the wall and allow these cells to enter. And we're not going to know that unless we can accurately measure these cells in the tissues and figure out whether, in fact, they are resistant to being cleared. And so this team of investigators has really been working on identifying these cells in the tissues. They've also been focused on another type of tissue-resident cells, the aptly named tissue-resident memory cells. This is another group of T cells that are important to study. As most CD4 T cells may freely circulate between the blood and the lymph nodes, these specialized tissue-resident memory cells remain in the gut and other effector tissues in the body and don't circulate in the blood. And again, we can't measure these cells by getting a regular blood draw. Tissue-resident memory cells may also preferentially support latent HIV infection, and this may allow the virus to hide from the immune system and avoid being killed. And it turns out that the very mechanism that retains these tissue-resident memory cells in the tissues may be contributing to HIV latency, or that silent form of HIV infection where it's not producing new virions. The tissue-resident memory cell gets a signal from the epithelial cells in the gut, which suppresses the reactivation of HIV provirus. So new virions are not made, and it remains silent and hidden from the immune system. That same signal causes expression of receptors that keep it bound to the epithelium and retained in the gut. So the very same mechanism that keeps it in the gut is suppressing the virus. And this may lead these cells to escape killing by the immune system. They may represent a unique barrier to curing HIV infection. And so this team has really been working to measure the amount of HIV in these cells, and we can now isolate these cells from gut biopsies and lymph node biopsies and measure the amount of HIV in these cells. So that we can now do this in the context of future studies in HIV-infected participants in our research studies. And one of the things that we plan to do in the upcoming year, you'll hear from Dr. Pillai and Dr. Deeks in upcoming talks about an analytic treatment interruption study, where we're trying to see whether there are particular measurements that we can make of the reservoir that predict the time it takes the virus to come back once HIV medicines are stopped. And we think that these tissue-resident cells that harbor the virus may be important predictors of the time it takes for the virus to come back, since they may be really important reservoirs. We also, in the more distant future, hope to assess the impact of shock and kill strategies like TLR stimulators that Paul mentioned on these cells to see whether these are more difficult to clear than cells that we may get in the blood, and whether we need new interventions to add on to our therapies to improve things. The next thing the module is working on is characterizing the precise anatomic location and phenotype of cells within the tissues. So what I've talked about are strategies where we can extract cells from the tissue. We want to look at what's going on in the tissue itself. And this team led by Zoltan Lasik with Joshua Vasquez, Raja Hussain, and Brandon Aguiar have really been working hard on developing methods to assess HIV right in the tissue itself. And this is actually some of the first data that any group has done so far in people living with HIV, where we're able to actually identify the cell types that are actually expressing virus in the tissues. Let me show you some of those data. So this is a lymph node from an untreated HIV-infected individual, and I recognize some faces in the audience. There are some people who, here who may have volunteered for research studies in the past, and many people who may have even undergone a lymph node biopsy. This is the type of data that we can get from lymph node biopsies in the context of HIV. This white stain is actually for the virus itself, HIV RNA. It's genetic material. This represents active virus production in the lymph nodes in this untreated patient. And what we see are two distinct patterns of virus expression. One is a so-called cell-shaped pattern. The blue stain here is the nucleus, or the center of the cell. And surrounding that nucleus, blown up here at higher magnification, it's just surrounded, chock full of virus, HIV RNA. This is a cell that's actively producing new HIV virions. This is a producer cell. And you also see another pattern. It looks more like a lattice-like network. And these are actually virions outside of cells. These are virions that have been made and are now trapped on dendritic cells in a B cell follicle. And we can start to ask, can we prove this? That this is in fact in the B cell follicle and not some other random place in the lymph node. Well, we can use a stain for CD20, which stains B cells in red, and we can see that same place where we saw that lattice-like network of virus in the tissues corresponds to exactly the same place where we see the B cell follicles. And you'll note that cell that was producing lots of virus is obviously not a B cell. It's not staining positive for CD20. But that cell, on the other hand, is staining positive for CD3, the T cell receptor. This was a T cell, as expected, that was producing all that virus. And we can now put all those stains together and get a complete picture of what the B cell follicle, the location for where the virus is being expressed and then retained in the tissues on that dendritic cell network, and also the identity of these HIV-producing T cells all at once. And the team is now moving on to define, with specialized stains, to define those tissue-resident memory T cells and also T follicular helper cells, the specialized T cell subsets in the tissues, to better define which cells are producing the virus and exactly where in the tissue. And again, this is the first time a group has actually put all these stains together. Others have measured the virus in the tissues, but not really identified the nature of the infected cell, at least in people. And so the final team I wanted to share with you is the team led by Nadia Rowan with Jason Needleman and Karen Jang. And they're really developing methods to more accurately identify signatures of latently infected cells and tissues. And why is that important? Well, if we can identify signatures of cells that are silently infected with HIV, it may give us some insights and ways to actually target those cells biologically, but we can also use that information to pair with imaging techniques that Paul referred to earlier and that Satish will talk about later, that may allow us to track these cells over time without even doing biopsies, which would be really amazing. And so what are we doing and how are we approaching this problem? Well, currently we only have a few markers that can give us a sense whether a cell is likely to be latently infected or enriched for HIV. There are some so-called exhaustion markers that are often expressed on cells that are enriched for HIV, so-called PD-1, LAG3, TIGIT. But the problem with these markers that we have now is that they're not very sensitive, meaning that many HIV-infected cells don't express these markers at all. So just looking at these cells is probably going to miss many of the latently HIV-infected cells in the body. It's also not very specific. So most CD4 cells that express these markers are actually not HIV-infected. So we may not be measuring the right cells if we limit ourselves to just these three markers. And in many ways, it's like trying to describe a face by just the presence of an eyes, a nose, and a mouth. You can't really describe someone's face by just the presence of those things. Rather, what we really need is like facial recognition software for the HIV reservoir. We need multi-dimensional descriptions of the surface of those cells to really get a sense of what those cells look like. And Dr. Rohn's group is now developing a CyTOF panel. This is a novel technology that allows for multi-dimensional phenotyping of T cells. And her group has generated a 38-panel parameter CyTOF panel to provide really high-resolution phenotyping of these cells. Not just 3 markers, but 38 markers to get a full characterization of these cells. And what's more, using a novel technology invented at Stanford, her group is adapting this to actually measure in the same highly phenotyped cells probes for detecting HIV expression. So we can say which of these cells are actively producing virus both before and after stimulation. And it's after stimulating the cells that we can flush HIV out and figure out which cells those are. Now, one of the problems of this approach is that HIV reactivation can alter the cellular phenotype. And so our lady who is doing the facial recognition software may have tried to disguise herself by putting on a fake mustache, right? This is something that HIV may do. It may alter the appearance of the cell. But the facial recognition software, or the CyTOF measurements, will help us to identify the original phenotype of the cell. Like such. And we can use fancy statistical techniques, in this case called the nearest neighbor analysis, to calculate what the face would have looked like had they not had the mustache, or those markers that HIV had caused to be expressed on the cell. So these are the techniques that we're pursuing now in the context of the charting module. And we look forward to carrying these forward in the upcoming year. And with that, I'll finish, and Kevin can bring up the next video.
Thank you. So one of the things that comes out, I think, sometimes in these talks that you probably grasp very quickly is that there's a lot of questions. Not so many answers yet. A lot of questions, a lot of things that we don't know that we're trying to understand through this research. The next video, and I think you will also see that there is some redundancy, naturally, in these videos because they are meant to stand on their own for people to be able to look at each of them individually and understand them. But the next video approaches the question of why these reservoirs continue to persist in the way that they do. Why do they remain in a latent stage? And that's the that's the understanding the reservoirs. To address that, after the video, Dr. Warner Greene, who is also in your program, so you can learn about him there, but represents, I think, as all of our speakers represent today, just the extraordinary talent here at UCSF and why we chose UCSF for the amfAR Institute. Dr. Greene represents, I think, one of the brightest, most creative scientists in the world today, and I'm often humbled to be around him and learn from him. So we'll watch the video, and we'll learn from Dr. Greene.
The main obstacle to an HIV cure is the reservoir of virus that is able to stay under the radar, evading attack by the immune system and powerful anti-HIV drugs. Understanding how the reservoir is formed and maintained is a critical step on the path to an HIV cure. The HIV reservoir consists of viruses that are integrated into the cell's DNA, but unlike in most infected cells, these viruses do not make copies of themselves. How do these viruses become latent when most other viruses are actively replicating? One hypothesis involves HIV infecting T cells that were previously active, but are changing back to a resting state. Once the cell is resting, the HIV inside it also rests and becomes latent. Some scientists think that the reservoir could also result from the virus inserting itself into a section of a person's DNA that is not very active. The virus isn't able to replicate in this inactive region of the DNA, but may be able to in the future. Another hypothesis is that there is an increase in the activity of proteins that block HIV replication, leading to latency. Once the reservoir is established, how is it maintained? Why don't the reservoir cells simply die out over time? Reservoirs could be maintained by normal ongoing cell division, a process that enables cells to replace themselves before they die. When the cell divides, the resulting two cells each carry a copy of the provirus. Sometimes, viral replication goes awry and results in the production of viral proteins that are not themselves infectious. But this can create an environment that activates T cells, giving HIV more targets to infect. In other instances, HIV infection has a disruptive effect on the gut, causing bacterial product to leak out. This leads to inflammation and also creates more target T cells for HIV to infect. This constant activation of the immune system can also exhaust it, preventing it from mounting an aggressive response to HIV. An immune system that's too exhausted to respond does not effectively kill infected cells, and the reservoir is perpetuated. By understanding the role these hypotheses, and possibly others, play in sustaining the viral reservoir, scientists hope to determine how to disrupt latent viruses and clear the reservoir. Join the countdown to a cure for AIDS.
Sorry. I have to say, I think these amfAR videos are spectacular, of high quality. I plan to use them as teaching aids, for sure. Thank you all for coming here on World AIDS Day. And I think just before I begin my presentation, I'd like to reflect with you for just a moment, my own personal reflection. I remember when I arrived in San Francisco in 1991, almost 25 years ago, almost to the day. And I remember starting attending on the wards at San Francisco General Hospital. Every patient in that first month had AIDS, and there wasn't a thing we could do about it. Now we've come so far down that road, but still the journey is not done. We do not have a cure. We do not have a vaccine. So the amfAR Institute for HIV Cure Research is designated or devoted to making sure we take care of one of those two goals. So my charge today is to talk about the understanding module, how effective these toll-like receptor agonists are in their ability to attack the reservoir. So let me tell you a little more about this reservoir to give you a sense of what it's about. So the best characterized reservoir, it may not be the only reservoir, but the one that we know the most about is the one that sets up in these memory CD4 T cells. And as the video showed you, these cells are very, very long-lived. But the number of the cells that become latently infected are quite few, one in about 1 million. So let me put that number in perspective. Take all the people that live in the Bay Area, 4 million people. The number of people that would be latently infected, there might be one in San Jose, one in Oakland, one in San Francisco, and one in San Mateo. And our goal, or our challenge, is to be able to study those 4 cells amongst the millions and millions of other cells. And our further challenge is to be able to attack those cells and kill those cells without harming any of the other cells. So we are up against a hard numbers game, and we need better and better tools to accomplish our goal. Now, the good news is the reservoir isn't very large. There aren't a huge number of these cells that have to be eliminated. But as we could see from the Boston patients and probably the Mississippi child, you've got to get every last one of those cells or the virus will bounce back. And finally, because of their long survival and the ability of these memory cells to proliferate, essentially, once you establish the latent reservoir, and it's established within days of HIV infection, once established, the reservoir is there for life. So, the one question is that the field has struggled over, is the latent reservoir an accident, or is it actually a planned event? The prevailing view until recently was it's just an accident. It's an accident as described by Bob Silicano. It's an accident of cells becoming infected, of memory cells becoming infected as they retreat back into that resting state. And at just the right time, the virus goes latent as the cell goes into rest. And what better place to set up shop, to hang out for long periods of time than in a memory T cell, which is hanging around, of course, to respond to antigens throughout our lifetime. And the virus takes advantage of that cellular environment, but the notion was it's an accident as the cell retreats into its resting memory state. So that's the accidental model. Contrast that with studies that have been reported in 2015 by Lior Weinberger at the Gladstone Institutes. Lior says, no, this isn't an accident. This is a highly planned event. It's hardwired into the virus to establish this latent reservoir. And in fact, it is a mechanism by which the virus can survive the rigorous challenge it faces when it infects a mucosal surface. Indeed, the virus it's a tenuous beachhead at best, and establishing a latent state may be a bet hedging strategy used by the virus to survive, for that cell to survive long enough to get into a lymph node and spread the infection. So two very different models, one highly planned and one accidental. It'll be important to distinguish between these two models as we move forward because they have implications for how we try and prevent the reservoir from being formed in the first place. Any hope for an HIV cure? Of course, what is driving this field is the success with Timothy Ray Brown. As we've heard, we succeeded. Doctors in Berlin succeeded with 2 stem cell transplants. He's now had no detectable virus found since 2007, and he's considered an example of viral eradication. Our challenge now is to go from N of 1 to N of stem cell transplants. So we need an alternative. But as a proof of concept, Timothy Ray Brown certainly drives the field, knowing that it's possible to get across that goal line. We just have to figure out better, smarter ways that are more effective. And in that regard, an HIV cure must be safe. Antiretroviral therapy now is quite easy. It's easier to take than it used to be. The cure has to be very effective. It has to work in a majority of the individuals who take it. And of course, the cure has to be scalable. We have to be able to use this in parts of the developing world where the virus is hitting the hardest. What will our approach in the amfAR Institute be? Well, you've heard a bit about it in terms of the toll-like receptors. These are the individuals, the scientists who are involved in Module U. I will not introduce each of them other than by their names here in these pictures. A very talented group of basic scientists, both in academia and in industry. The slide advances about 1 out of 4 clicks. Okay. All right, so we will try to harness the innate immune response for the ability to shock and kill the latent reservoir. We want to try and turn this normal innate immune response into a killing response, but we only want to do it in those 4 individuals that are in San Jose, Oakland, San Francisco, and San Mateo, not everybody else. So this is a picture. You probably recognize this immediately as a toll-like receptor. This is the membrane. It's an elegant structure. It's a dimer. There's 2 of them. And this is the toll-like receptor agonist. And these are we're changing, these are the activators that cause this dimerization and the activation of the receptor. And we're changing these agonists so that we can activate different types or different flavors of toll-like receptors. It was in 2011 that for the discovery of the toll-like receptor innate immune system that Bruce Beutler and Jules Hoffman won the Nobel Prize. So this is an overview of the toll-like receptors. Lots of lollipops that exist in dimers. But interestingly, the point I want to make here is some toll-like receptors are designated to respond to be an early defense against fungi, others against bacteria, and these against viruses. And of course, we've had a lot of focus on the ones that maybe stimulating these with our agonists will create a better viral, antiviral response. But in fact, it turns out that some of these toll-like receptors that are designated for bacteria, that they can be stimulated as well and harnessed toward creating an anti-HIV response. So the first question was, how are these toll-like receptor agonists working? We had we had some very nice agonists from our colleagues in Seattle, the Infectious Disease Research Institute, against TLR4 and TLR7. The notion was that while TLR receptors, toll-like receptors, exist on T cells, those receptors might not be all that functional. But we wanted to understand, in the case of HIV, whether or not, if we added these agonists on purified CD4 T cells, whether we would get any kind of responses. And so we essentially created latently infected cells, and then 6 days later, we treated them with the toll-like receptors, and then we checked for viral reactivation in these latently infected cells. And what we found was a monotonous lack of response in the purified CD4 population. There was no toxicity of the toll-like receptors as evidenced here, but there was no response. This is an example of one donor. These are studies that were performed by Grace Shi in the laboratory at Gladstone. So we moved on to then say, okay, if it's not acting directly on the T cell, might it be acting on an intermediate cell, for example, a dendritic cell or a macrophage? So the idea would be that the agonist could work on these toll-like receptors that are present on these dendritic cells or macrophages. These cells could in turn produce cytokines that now activate the CD4 T cell harboring the latent virus. If that's the model, we sure as heck want to know what that intermediate cytokine is, because we could also harness that for our purposes. I think I understand the problem. I think the battery is running low because my pointer has failed as well. Okay, so what we did was now to set up culture systems similar to the last one, except we supplemented these culture systems with dendritic cells. And now we are stimulating with the toll-like receptors in the presence of latently infected T cells and dendritic cells, and we got quite a different response. As you can see here, with TLR7, 8 agonists and TLR4 agonists in the yellow and kind of polka dot red, we see that there was a response, about a 3-fold induction of virus production in this particular donor. So this was, now I have to say, this is a response. It's clearly reproducible, but this isn't a great response. This is not a 100-fold stimulation of virus production. So I would conclude that this series of studies that were performed, that there is a consistent, although moderate activation of latent virus expression when monocyte-derived dendritic cells and latently infected tonsil cells are co-incubated together and stimulated with activators of TLR4 and TLR7. But we didn't hit it out of the park with this study. Any synergy or additivity? Well, of course, we're very interested in that. Could we combine 4 and 7 and get a better response? Well, nah. As shown in the yellow bar, it's not even additive. It's not, you can't take the 4 and the 7 response and put them on top of each other. The yellow bar is less than that. Certainly not synergistic, probably not even additive. So, one agonist is as good as adding 2. So no evidence for clear synergy or even additivity when the 4 and the 7 activators are combined. Now, So how active are these TLR agonists when we use them not in this model system, but in the gold standard? The gold standard are cells from HIV-infected individuals who are on long-term ART. So we are very fortunate to have many volunteers from the community who are willing to share their blood with us by leukapheresis. And it is these types of precious, precious cells that form the gold standard for our studies of HIV latency. Let me show you some of the results that Grace generated. So these are the HIV-infected, latently infected cells from latently infected cells from patients on ART. We see, in fact, no response, hardly any response to TLR2 and TLR3, but wow, a 12-fold response on day 4 to TLR4. What I'm showing you here is the best response that was obtained over 3 doses of each agonist and over 3 different time points of cultivation. So there is a good response to TLR4, a modest response to the TLR7, TLR8, and 7/8 agonists, and interestingly, TLR9 comes into play. This is recognizing DNA. So these primary cells from HIV-infected individuals were started to show us more promise than we saw in the model system. But still, no question, we conclude that overall several TLR agonists exhibit moderate shocking activity in cells from HIV-infected individuals on ART. However, these agents appear more active when they're given in vivo. Even in animal models or in humans, these agents appear more active in vivo than they do in the test tube. And it's likely because in vivo, these toll-like receptor agonists not only are serving as shocking agents, but the purpose of the toll-like receptor system is to provide that early warning, early defense, but also to get the adaptive immune system, the antibodies, the cytotoxic T cells, the real fighting arm of our immune system on board and ready, primed, and ready to go. These toll-like receptors actually act as adjuvant signals, boosting the adaptive immune system. So we don't model that in vitro, but in vivo, we're seeing really interesting effects of these agents. So I would like to conclude with just a little bit of original science as well from our module. And I term this putting the kill in shock and kill. So the idea, of course, with shock and kill is to use a shocking agent to stimulate this latently infected cell to now make itself immunologically visible. Actually, the idea, hopefully, is to make it produce so much virus that it dies a natural death within a short period of time. And the idea then, of course, is to protect so the virus coming out of this cell before it dies, it can't infect any other cell because we're doing this under the protection of antiretroviral therapy. Now, what is emerging now is that we never, with our shocking agents, induce enough virus to cause this cell to spontaneously die. As such, it's likely that we're going to have to engineer into the system a killing function, that somehow we're going to have to encourage these cells, either by cytotoxic T cell mobilization or antibodies or other approaches. We have to trigger these cells to die by a second step. Let me show you a novel second step that was created by, that actually involves harnessing a second component of this early defense mechanism, the innate immune response. And these are studies that were carried out by Pei-Lin Lee and Joe Wong and recently reported in Nature Medicine. And what Pei-Lin and Joe did was they took advantage of a biological defense system called RIG-I, RIG for retinoic acid inducible gene 1. And actually, what they found, they took a drug that is already FDA approved, that is used in the treatment of psoriasis called acitretin. One of the functions of acitretin, it's a retinoid inducible gene 1. So acitretin can actually induce new expression of RIG-I And interestingly, acitretin has some weak latency reversing activity, the ability to wake the virus up out of slumber. But the really key part is that with the induction of RIG-I, now if the virus is viral RNA is being expressed, that viral RNA can bind to the RIG-I and activate a cell death pathway that causes that cell selectively to die. So it's a very fascinating type of model that works beautifully. This is the pathway, ultimately leads to death as well as to interferon production. But what they showed is when they took these cells from HIV-infected individuals on antiretroviral therapy, which has not been shown with any other latency reversing agent yet, is that acitretin was able to reduce the amount of HIV DNA, and by analogy, the number of latently infected cells in those cultures, and it got even better when you combined it with other latency reversing agents. So these are exciting first studies, all in the test tube at this point, but they are now driving a clinical trial, a proposal for a clinical trial to test acitretin as a way of trying to mobilize a death pathway to selectively kill the reservoir cells that have been reactivated to express viral RNA. I believe that's this was just a summary slide describing that, and certainly it's been a thrilling ride to see Joe and Pei Lin develop these studies. Thank you very much for your attention.
Well done. I suspect that many of you, like me, are asking the question right now, is Dr. Greene right? Are the batteries on this thing really low? I'm not sure I know the answer to that, but maybe we could get it checked out in case it is. Yeah. You have new batteries? Here, why don't somebody come get this? I want to remind you about the cards that are in your seats. If you have a question, write it down on the card, and some folks are going to be coming through the aisles now. Raise that up above your head, and they'll pick them up from you. Because after we finish all of these presentations, that's when we're going to be bringing all of the panelists up here, and I hope having a very sort of robust conversation and maybe even debate at some point. So our next video is the R, record. It's about figuring out how big these reservoirs are. And Dr. Greene mentioned that we have some information about the size of these reservoirs. I thought it was interesting, the analogy of having, a person in San Francisco, one in Oakland, and one in San Mateo. I would add to that analogy only one piece, and that if there are 4 we're looking for, we know about the one in San Francisco, we know about the one in Oakland, we know about the one in San Mateo, but the other one we don't know where it is. We just know it's in the Bay Area somewhere. And that's how hard it is to figure out the question of reservoirs. We know they're there, but sometimes it's really, really difficult to sort that out. So this next video is going to talk about measuring the size, and then I'm going to bring up my favorite geek, Satish Pillai. And Satish and I, we were at a meeting several months ago of one of our amfAR's big donors, and I got to introduce him, and I introduced him as my favorite science nerd. But he's actually really the coolest geek I know, which is not a very high bar, but nevertheless, I think he really is terrific and very, very bright, and you're going to get to see that in just a second. So let's run the next video on recording the reservoirs.
The main obstacle to an HIV cure is the reservoir of virus that is able to stay under the radar, evading attack by the immune system and powerful anti-HIV drugs. Measuring the size of the reservoir is the third critical challenge standing in the way of a cure. During the course of its life cycle, HIV can become defective to varying degrees. If it's sufficiently defective, it may never have the potential to cause harm. This type of benign provirus may not need to be eliminated. we need to know how much virus is left that does have the potential to cause harm once the person stops taking ART. Intact provirus results in infectious virions and is obviously an important contributor to the reservoir, propagating infection in the absence of ART. However, even provirus that is so defective it cannot result in an infectious virion can still be harmful. These defective proviruses can cause inflammation and wreak havoc on the body. Recording which proviruses fall into these categories, benign, infectious, or inflammatory, will give us a clearer picture of how many must be eliminated. Most importantly, by being able to precisely measure all of these types of proviruses, we can determine whether a curative intervention is working and effectively eliminating the reservoir. Join the countdown to a cure for AIDS.
Awesome. Hi, everybody. It's my distinct pleasure to be able to talk to you on this very auspicious day. And after Kevin's introduction, I'm not sure, am I cool or not? I'm not sure. One of the two. But I'm here to talk to you today about recording the HIV reservoir. And I think very importantly, I want to start by introducing our team, because I'm definitely here as a representative of a team of just absolutely fabulous, brilliant, collaborative, friendly scientists. I'm not going to go through everybody name by name except for when I introduce their contributions to the data that I present today. In particular, I want to highlight something that Warner also mentioned, which is that many of the people here, so everybody in the top row and a few of the people on the bottom, are based here at UCSF. But both Mike and Chris Petropoulos are based in the private sector. They're in industry. And I think one of really the critical strengths of our institute here and our approach is we're very heartily fused with the private sector, which kind of brings a unique strength and inertia to what we're doing here. So again, I know I'm risking being slightly redundant, but I think it's really critical to go back to the fundamentals of HIV persistence really briefly to frame the approach that we're developing here and what I'm presenting to you. So if we think about untreated HIV infection, so no antiretroviral therapy on board, this is how HIV persists. I think it's pretty straightforward. Imagine that you have a large population of HIV-infected cells. Those cells are going to produce virus. The virus will travel around the body and encounter uninfected cells. Then the virus will infect those cells and generate a whole new population of infected cells. And this whole process will continue ad nauseum. So this is how the virus persists in the context of untreated HIV infection. Now, when we bring antiretroviral therapy into the picture, we completely shut down the right half of the graph here. There's no more replication, so there are no new events that are being colonized by HIV. But what we do still have are a population of latently infected cells. So again, antiretroviral therapy is blocking new cells from being infected, but it's doing absolutely nothing to get rid of the population of cells that were already infected before it got there. And what we're learning is that a lot of these cells that harbor HIV can live for a long, long time. We don't know how long. They could live for months and maybe even years. And so that's the challenge that we're dealing with. So why are we interested in counting these cells? Why are we interested in recording the size of the HIV reservoir? So I think we're all familiar with the basic natural history of HIV. This is the pattern that we almost always see. So on this axis is viral load. When we place an individual on antiretroviral therapy, their viral load drops below the level of detectability. So this dashed line here represents a sensitivity of the usual methods that we use in the clinic to measure HIV. So once we start antiretroviral therapy, the virus drops to undetectable levels. But what we're all too familiar with is when we stop antiretroviral therapy, the virus almost always comes back. It's pretty much a given that the virus will come back. When it comes back, however, can be fairly variable. But you can be pretty sure that the virus will, in fact, come back. Still isn't functioning very well. So ultimately, this is what we're trying to do in the module that I'm leading. We're trying to develop a new generation of tools that allow us to predict if and when the virus will rebound after we stop antiretroviral therapy. So how can we actually go about doing that? The one thing that we know is that if we study several different individuals who are on antiretroviral therapy, there's dramatic differences in the size of the latent reservoir between individuals. So what I basically mean is that some individuals have lots and lots and lots of latently infected cells, while other individuals tend to have a smaller population of latently infected cells. So in the scenario that I'm presenting here, an individual with a large reservoir, I have very meager art skills. I was trying to compete against this is literally like my neck-and-neck competition with my toddler daughter, and I still lost, actually. But basically, this here is supposed to represent an individual with a large HIV reservoir. The virus will come back relatively quickly. But what we know from a lot of different studies at this point is that when an individual has a smaller latent reservoir and just maybe a small handful of latently infected cells, the amount of time that it takes for the virus to return after antiretroviral therapy is stopped is much longer. And so really the goal of this module is to develop accurate, sensitive, reproducible, and scalable strategies to be able to gauge the size of the latent reservoir. So where are we right now? This is kind of how I picture where we are right now. I think we desperately need a revolution in the area of HIV reservoir recording. So I encountered this ad. This ad is from High Fidelity magazine from sometime in the 1970s. And this is kind of where we're at, I think, now with reservoir recording. We're sort of in an era that's pretty crude. We have a lot of very expensive technologies. They all do slightly different things and tell different stories. But we don't really know if any of these things are actually telling us the absolute truth. And I was thinking about this morning. I think now that I showed this, there are probably going to be some real diehard analog recording fans that are going to send me like hate mail. Actually, I'm one of those people. I'm going to send myself hate mail. But really what we need are a new generation of modern tools that can accurately measure the reservoir in a cost-effective fashion. So one thing that we really need is we need more sensitive recording tools. So what do I mean by that? There have been multiple cases now, including the famous case of the Boston patients who were managed by Dr. Timothy Henrich, who is a big part of our Cure Institute. What we found there was the virus rebounded in these individuals. Several different labs had tested samples from these individuals and could find no conclusive evidence of any remaining viral nucleic acids in those samples. Yet the virus came back. And so the take-home lesson there is that the virus was lurking all along, but the methods that we used to measure the virus in the clinic were not sensitive enough to zero in on the residual virus that was in the individual. So we need better, more sensitive tools. So here's really, that leads me to my first challenge, I think, associated with the tools that we're trying to develop. HIV persists at extremely low levels during antiretroviral therapy, which really is kind of a classic representation of the needle in the haystack problem. So, yikes. What was that? Everybody still alive? So just to give you some numbers on how rare these cells are, this is work from my lab where we've measured the latent reservoir in a large population of about 72 HIV-infected individuals on antiretroviral therapy. And so on this axis right here, I'm not sure my laser pointer is working. Okay, there it is. On this axis right here, this is a representation of the frequency of HIV DNA and CD4 T cells. And we divided our cohort into individuals that started treatment during early infection, like within the first few months of infection, versus individuals who started treatment during chronic infection. So one thing that you can see is there's a significant increase in the size of the latent reservoir in individuals who started treatment later. And this is a well-substantiated fact. Many different groups have reported the same detail. Another thing that we tried to look at was these colored dots represent really interesting human genetic mutations that are known to affect HIV disease progression in the setting of untreated disease. We wanted to see if there were any correlations between these well-recognized human genetic mutations and the size of the reservoir. Long story short, we found some associations, but they were modest at best. But the real interesting thing, I think, is if you look at these dots down here, this is a log scale, so 10 to the 1 is actually 10 copies. So what you can see is in a lot of individuals, you find as low as 10 to 20 copies of HIV DNA in 1 million CD4 T cells. Again, driving home the point that these are very rare targets and we need extremely sensitive tools to zero in on the virus that persists. So this is, in a nutshell, the strategy that we're using. And I actually presented this graphic last year, and I want to show you a little data that we've generated over the course of the last year. Our strategy, in the nutshell, is we think we can do a better job at finding the needles in the haystack by just sampling a larger portion of the haystack. So the current methods that are used in the laboratory to find viral nucleic acids generally can characterize 100,000 cells or less at once. We're using this digital droplet technology where we can pump in 1 million cells or more. And actually, we're kind of continually ramping this up. So it looks like we can actually start with millions of cells to look for 1 or 2 viruses. And I think this will give us a significant advantage and should give us a much greater probability of encountering any virus that persists. So this is how we actually test the sensitivity of our assays. So we start with a whole bunch of uninfected primary cells. So these are, again, human cells from an individual that is not infected with HIV. Then we have these special cells in the laboratory that each contain exactly 1 copy of the HIV virus. And we spike these infected cells into the background of uninfected cells in known proportions. Then what we do, before I walk you through this scary-looking plot, we essentially break the cells open, we take all the DNA out of the cells, and we chop the DNA in a blender, and then we suspend them in millions and millions and millions of droplets. So at the end of it, we have tons and tons of tiny little droplets with little bits of DNA scattered in them. And this is what the droplets actually look like. Each one of these blue dots represents a distinct droplet, and the droplets fall into 4 different character states. In the lower left corner, these droplets are just empty droplets, so you can just think of them as drops of water. There's no DNA of any sort in here. Up here, these are droplets that contain human DNA. So in this case, we're counting copies of the CCR5 gene. That's our marker of human DNA. Over here, this is the interesting quadrant. Down here in the lower right corner, each one of these droplets contains a single copy of HIV DNA. And then just by chance, you'll have some droplets that contain multiple copies of DNA, so they'll contain both HIV and human targets. So in this example that I'm showing you here, we started with 1,000 HIV-infected cells, and we spiked them into 1 million uninfected cells. And the proportions that you see here perfectly mirrors our expectations. So to put some quantitation on that, so we've spent several months over the year doing lots and lots of these experiments to see how robust, reproducible these assays are, and we wanted to see what the dynamic range was. So on the x-axis, this is the copies of HIV DNA that we're expecting. And again, unlike a patient sample, we have the luxury of knowing what the absolute truth is here because we engineered these cocktails in the laboratory. So we know how many copies of HIV DNA are in here. Up here, these are the actual number of copies that we recorded using our molecular tool that we've developed in the lab. And what you can see is there's pretty much a perfect concordance between the number of copies that were spiked into the sample and the number of copies that we actually registered in our assay. The other thing you can see is, there are these little error bars. Down here, when you get into the range where you only have, 1 or 2 copies of HIV DNA, there's some variability. But any time you get above, let's say, 10 copies of HIV DNA, there's almost no variability whatsoever. So, again, is this a perfect assay? No, I think there's still room for improvement. But we think that this assay that we've developed in conjunction with Raindance Technologies represents a meaningful advance over some of the existing tools that are available today. So I think this is a good time to segue into some work that built off of the digital PCR strategy. So this is Dr. Mohammed Abdel Mohsen at BSRI, who's an absolutely fantastic molecular biologist. So we are now using this digital PCR strategy to understand how HIV cure strategies, and in particular agents that reverse HIV latency, affect both the host and the virus. And as Dr. Greene mentioned, I think it's becoming patently clear that we're not just trying to reactivate the virus, because that's probably not going to be enough to result in the death of the cell. As we reactivate the virus, we also need to promote antiviral immune mechanisms in the host that are going to accelerate the destruction of the infected cells after the virus has been reactivated. So if we can design tools that allow us to measure both sides of the equation simultaneously, that's a real key advantage, I think. So this is what Mohammed and our group, this is what we did. So again, this is kind of an ugly figure, but I'll walk you through it briefly. These are just unstimulated CD4 T cells, so we didn't do anything to them. We just took them and we put them in a petri dish, essentially. When we look at everything in this circle, these droplets have no DNA, and in this case, no RNA whatsoever. P21 and ApoBec3G are 2 very important antiviral, anti-HIV immune proteins. So the number of dots that exist in this oval and this oval represents the expression and activity of the P21 and ApoBec3G antiviral genes. GAG, represented in this circle, the number of droplets in here measures how active the HIV virus is in itself. GAG is an HIV gene. So here we're cataloging the gene expression and transcription of the virus. And again, these are unstimulated cells. What we did was we treated these cells with a human sugar-binding protein called galectin-9, which we had some reason to believe would actually promote the reactivation of HIV. And sure enough, what you can see here, if you look at this gate, this gag gate, you can see that the number of droplets rises by about 20-fold, which means that the administration of galectin-9 very significantly promotes viral reactivation. What we can also see here is if we look at these gates for P21 and ApoBec3G, we see a profound elevation in both of those sectors as well. So if you put this together, our multiplex digital PCR strategy allowed us to take a potential HIV cure drug, treat cells in vitro, and determine that this galectin-9 sugar-binding protein could potently reactivate latent HIV and simultaneously induce key host immune defenses against HIV as well. And these data were featured in an article in PLOS Pathogens, which is one of the first few papers that have come out from our institute since its inception. So here's another key challenge. I already talked about the fact that HIV is very rare. The second key challenge, and I think a very intellectually interesting challenge, is that most of these viruses are genetically defective and cannot grow. So the vast majority of viruses that we encounter are incapable of replicating and are therefore, we would think, completely irrelevant to the likelihood that the virus will come back when we stop antiretroviral therapy. Why is this the case? Well, HIV has a very high mutation rate that enables the virus to escape immune pressure and also allows the virus to evolve drug resistance. But this really comes at a cost. There's no free lunch, right? The fact that the virus does such a sloppy job at reproducing itself results in the fact that the vast majority of viral genomes are littered with these mutations that render the virus totally inactive. And I just wanted to show you some data from our group that kind of describes this in a nerdy, geeky fashion. This is a phylogenetic tree of HIV genomes. And all of these viruses, we used next-generation sequencing technology to characterize viruses that were infecting a single individual. So each one of these branches in this tree that you see represents a distinct viral genetic variant that we cataloged in this individual. And so what you can see is, at any given time, there's this really, really diverse swarm of viruses in a patient, but only maybe a handful of these branches that you see on this tree are actually capable of replicating and reinstating infection once antiretroviral therapy is stopped. So really, I think a very simplistic way of thinking about this, and I think that this concept was highlighted beautifully in the video before my talk, HIV genomes can be classified into 3 basic types. So as pictured by this green light down here, probably the most important type are infectious replication competent virus. So the way I like to think about it is this sort of, very pristine-looking genomic DNA that's, colored like a candy cane. If only it actually smelled like peppermint or something, then we could use a peppermint detector. But since we don't have a convenient peppermint detector to identify replication-competent virus, we're developing several different novel strategies to be able to differentiate replication-competent infectious virus from genetically defective virus. So this nifty picture that I'll show you right here, Dr. Tim Henrich, who's playing an incredibly active role across the board in our Cure Institute, this is a microfluidic device that he's developed in his laboratory, and he actually just snapped this picture in his lab yesterday. And what this device does is it allows you to take CD4 T cells from an infected individual on therapy, and you can co-culture them in the microscale with feeder cells, and then you can actually see how often you have the outgrowth of infectious virus. And so this allows you in a microscale, hopefully cost-effective and reproducible fashion to gauge the total number of infectious viruses in a clinical sample. And there are methods that exist that do this, but on a very large scale off of a chip. They're called traditional quantitative viral outgrowth assays. But these assays are extremely laborious and very, very costly, and their reliability has also been called into question. So we think that this kind of micro version or nano version of the viral outgrowth assay could be a significant advance. So, okay, I mentioned that there's replication-competent virus. The other main type of virus that we're interested in are these benign, completely defective viral genomes, as pictured here. And this is just a genome that looks like it's totally thrashed and beaten up. So how are we going to specifically identify those defective viral genomes? Again, we're developing several different strategies, but the one thing that we've been working on hard over the last year is using, oh, the text got cut off there, but we're using a combination of the droplet digital PCR technology that I mentioned with next-generation sequencing to be able to characterize the exact genetic sequences of all the viruses in a clinical sample. And then we can use computational tools to determine which of the genetic sequences that we've characterized are likely to produce infectious virus and which of them likely represent dead ends that will not result in propagating infection after treatment is stopped. Lastly, there's this third class of viruses, and I think this is actually kind of a new chapter in the field of HIV persistence research. In addition to defective genomes and genomes that are replication competent, there's this third class of inflammatory defective viral genomes. So what these are viruses that probably do not have sufficient genetic integrity to actually replicate and grow out after therapy is stopped, but they're still a major concern to our health because they probably produce bits and pieces of proteins that are seen by the immune system, and so they can drive chronic inflammation. So even though they're not actually replicating and growing, they're still causing harm by promoting immune activation and inflammation. So I think the field is kind of awakening to this concept and now realizing that we're probably going to have to figure out what to do about this class of genomes as well. What do these genomes look like? I don't think we're sure, but I think we're going to hear a lot more about this class of viral genomes in the year ahead. So the final challenge that I'm going to present, which may be the most profound out of all of these challenges, it's becoming increasingly clear that HIV persistence in peripheral blood may not give us the information that we need to adequately gauge the efficacy of our curative interventions. We most likely need to drill deep and interrogate virus that persists within tissue environments, like especially lymphoid tissues. So how can we do that? It's obviously very easy to collect a tube full of blood from an individual. It's really hard to do serial biopsies of lymph nodes from individuals. That's clearly not a scalable strategy. So we need to find efficient and non-invasive modalities that we can use to sample virus within the tissue environment. So one of the concepts that we're kind of building our projects on is this, that the host immune response against HIV may give us indirect but critical information about the size of the latent HIV reservoir. And so this is one example. Circulating antibodies against HIV that are present in the blood may give us key insights about HIV persistence in tissues. And the thinking is like this. These HIV antibodies are circulating throughout the blood, and they're going in and out of tissues. And so they're probably touching bits and pieces of viral antigen that are remaining in pockets within these tissues. So when we sample these antibodies in blood, they probably can give us some information about their experience within the tissue environment itself. So I will show you a little bit of data that we've generated in the last year. This is from Mike Bush and Sheila Keating, where they've been able to demonstrate on this axis here, this weird-looking thing, log-lag ODN, just refers to the concentration of antibodies against HIV in the blood. And what they could show was there was a very solid relationship between the amount of circulating antibodies against HIV that they could find and the amount of HIV RNA that they could find in that patient's cells. And so this really lends credibility to the idea that these antibodies give us a window into how much virus persists within the body. In addition, and I think this is really maybe a very important result, although it needs to be substantiated with much larger sample sizes, we studied the concentration of these antibodies in a small population of about 10 individuals who stopped antiretroviral therapy, and we waited to see how long it took until the virus came back. And what we could see was individuals that had lots of antibodies against HIV, this seems paradoxical at first, but individuals that had lots of antibodies circulating against HIV were the first to experience viral rebound when treatment was stopped. And individuals that had low levels of these antibodies actually went a fair amount of time before the virus actually came back. So I think this lends credibility to the idea that these circulating antibodies are telling us something meaningful. And we're hoping to explore this in a much larger cohort over the next year. So I think probably one of the most exciting things that we're working on is this idea of in vivo imaging. And a lot of this is being driven by Dr. Henry Van Broeklen here at UCSF, along with Tim Henrich and Priscilla Hsu. So our plan for using in vivo imaging to visualize a latent reservoir is based on this concept. We can take antibodies that are specific to the HIV envelope protein, and we can radio label them with something that glows. And then what we can do is we can measure the activity of that radio label using immuno-PET-CT. And so what we should be able to do is really we should be able to look throughout the human body to identify pockets of HIV persistence and HIV activity, which is totally unprecedented. This has never, ever been done in humans. This has only been done in monkeys at this point. So actually being able to specifically study the anatomic distribution of HIV in infected humans is going to be a major advance. So what have we done on this front? Dr. Van Broekelen has been able to obtain a broadly neutralizing antibody against HIV called VRC01 from the National Institutes of Health. And he's radio-labeled that with a radio label called zirconium-89. I have no knowledge of biochemistry, so this just looks like a bunch of squiggles to me. I can't walk through this myself. All I know is he took these antibodies and he did something really fancy to them, and he attached this radio label to them. And what he's been able to do at this point, in addition to radio labeling it, he's gathering a whole bunch of initial data to submit an investigational new drug application to the FDA, which they're anticipating to do in the next few months. So we're hoping that sometime in the next year we'll be able to actually take these highly specialized radio-labeled antibodies and measure HIV persistence throughout the human body. So in the past year, we've also done additional experiments using this in vivo imaging approach, although they don't involve directly imaging HIV itself. Instead of imaging HIV components, we've indirectly imaged HIV persistence by studying the metabolism of T cells. So in this case, this is a lymph node, and what you can see are there are no pockets of any activity. When you look down here in a lymph node from an HIV-infected individual, so this is from an uninfected individual, when you look in an image of a lymph node from an infected individual, you can see these glowing pockets here. So these pockets represent regions where you have uptake of this thing called FTG, which is a marker of the metabolism of a cell. So what this clearly tells us is that HIV, the presence of HIV, turbocharges the metabolism of cells in its neighborhood. And so this is one way that we've actually been able to indirectly image the presence of HIV using existing tools. Let's see here. This is just an example of the imaging roadmap in the years ahead. In addition to directly measuring HIV, Henry and Tim and colleagues are measuring the effects of HIV infection on a number of different metabolites. And I think over this roadmap, we'll have a very robust picture of how HIV persists in different tissue sites across the body and how it also affects inflammation and immune activation in its neighborhood. So I'm going to close with this figure, and this figure is admittedly quite manic because it represents a lot of different work, and I'm not going to go into it in detail. But I wanted to briefly explain what we've done in the last year, and actually even more than a year. We kind of got rolling on some of this work before the Cure Institute started, although it was funded by a previous amfAR grant that was given to my laboratory. So we're working full tilt to identify a biomarker of the HIV reservoir. So what we've been able to do in the last year or two, we've collected samples from 72 infected individuals on antiretroviral therapy. We've collected their blood plasma. Once we've had that blood plasma, we've measured HIV viral load, circulating anti-HIV antibodies, cytokines, which represent proteins that are involved in intercellular communication, exosomes, which represent these tiny little particles that are secreted by cells that are also involved in intercellular communication. Then we've collected their CD4 T cells, extracted their RNA and DNA. We've measured HIV DNA, and we've sequenced both the human DNA as well as the viral DNA. And lastly, we've taken their RNA, we've measured HIV RNA, which is an indicator of HIV transcription and activity, and then we've measured the expression of every human gene at the same time. So I'm not expecting all of this to make sense, but I kind of wanted to give you a peek into the magnitude of work that we've embarked on to really identify a reliable biomarker. And I just wanted to close with one image, which segues into Steve's talk. What we're really hoping to do, basically we have a lot of information from lots of different tools that we're developing in the lab, but we don't really have an idea of which of these tools or recording strategies is telling us anything meaningful. So the only way that we can determine if any of these tools that we're developing tells us something that's clinically usable is through this idea of an analytical treatment interruption. And as Steve will tell you, this year we are conducting an antiretroviral treatment interruption so that we can collect a large number of specimens right before antiretroviral therapy is stopped, and we can determine if any of the tools that we're developing can tell us how long it will take before the virus comes back. And really, ultimately, our long-range goal is, after a curative intervention, when the virus has been completely depleted, the recording tools that we develop should tell us exactly that the reservoir is depleted and the virus never comes back. So this is kind of the long-range vision that we're shooting for. And with that, I will stop and turn it over to Kevin. Thanks a lot.
Thanks, Satish. So someone has asked if you can access these videos and slides. As of today, the videos are going to be live on curecountdown.org. So you can go to that website, and the videos are there. And the slides will be going up as well, I think, shortly. We have permission to upload those. So you'll be able to access the slides and the videos at curecountdown.org. Last video and last speaker. So this last one is Eliminate, which sounds sort of self-explanatory, but to explain it for us, after the video, we have Dr. Steve Deeks, who, as many of you know, brings an infectious enthusiasm and a great deal of joy to his work every day. No, I'm kidding. He's a real curmudgeon. In fact, he describes himself that way. No, I like introducing Steve during the holidays, because if you've seen The Grinch Who Stole Christmas, you know I don't have to introduce him at all. So let's watch the last video, Eliminate, and then we'll have Dr. Deeks talk about it.
The main obstacle to an HIV cure is the reservoir of virus that is able to stay under the radar, evading attack by the immune system and powerful anti-HIV drugs. A cure for HIV will be achieved when the reservoir that can cause harm has been eliminated. We are gaining valuable clues about how to cure HIV from a small number of people whose immune system is able to control the virus without the need for antiretroviral therapy. We call this a state of remission. But those who are in remission require regular doctor visits to ensure that the virus remains under control and that they are not infectious. A cure, on the other hand, means targeting every provirus that has the potential to cause harm, those that are infectious and those that are inflammatory. This would allow a person to stop taking ART and to no longer be infectious. Importantly, if we could confirm that a person was cured, there would be no need for regular doctor visits. Several strategies are under investigation. One, called shock and kill, involves administering a drug that would shock the virus out of latency. Once the infected cell starts producing virus, the immune system may be able to kill the virus-producing cells. Other scientists are investigating whether gene therapy might be developed to, for example, cut the provirus directly out of the DNA. There are several steps on our path to a cure. We need to know where all the virus is located that must be targeted. We need to understand how the virus maintains itself so that we can devise ways to disrupt viral persistence. We need tools that allow us to accurately count every last provirus that can cause harm. Our goal is to eliminate the harmful provirus and achieve a cure. A few short years ago, very few people believed a cure for HIV was possible. Now we have proof that a cure is possible, and thousands of researchers around the world are working to make this dream of a cure a reality for the 37 million people living with HIV. amfAR is investing in all of these strategies as we work together on the countdown to a cure for AIDS.
So it's my pleasure sort of to try to end the formal presentations here. My job as a clinician at the county hospital is to basically link all the great basic scientists that you've heard from, who come up with all these wonderful ideas, with basically with the community here in San Francisco, which is very special. And I think I should just really mention what makes this community special. We have a large population of people with HIV. But more than that, we have a culture within this city that I think a lot of people with HIV tend to be very educated, very motivated, and often quite altruistic. And hence, it has been easy for me to do what I do, which is essentially take these ideas that are in the laboratory and move them into the clinic. And we do that primarily by doing early, often high-risk clinical trials, and I'm going to talk about a couple of them here, that are really important to stimulate the field to move forward. More importantly, equally importantly, when we do these studies in which we explore within people how we might actually cure HIV infection, we often, almost invariably, find out things that we didn't think was going to happen, which require explanation. And what this institute is able to do is to allow us to very seamlessly take the observations that we're making in these clinical trials with the specimens we're collecting right back into the lab to answer those questions as they come up in real time. And it's actually a synergistic relationship between the community, the clinicians, and the basic scientists, which I honestly think does not work as well anywhere else in the country, if not the world. And so I find myself with the scope team and the options team, many of the people here in the audience, sort of uniquely privileged to be sort of that link. So what I want to talk about in the next few minutes is just how we're going to sort of move some of the critical ideas from the laboratory into clinic, particularly this idea that we can turn on the immune system through TLR 7, which is sort of our key approach that we're taking strategically within the goal. But I will say up front, and so there's been some debate, Kevin mentioned this, and other people mentioned this, there's been some ongoing debate as to whether or not are we going after remission? Do we want to come up with a way to actually achieve control of the virus so people stop their therapy but it's still there? Or do you want to get rid of it completely? And I say we want to do both. But what we're going to do first likely is a remission. That's the first step. Because that's going to be easier to do than getting a complete and total cure. I actually think that's going to come next, after we figure out how to manipulate the immune system to control whatever virus is left. So what do we know about that? How do we control the virus? How do we envision that people who are on treatment today can go off therapy and not have the virus rebound? And we think this is going to be possible largely because it happens in nature. 1 in 100 people can control their virus on their own. These are elite controllers. And there's several people in the audience here who are elite controllers who have participated in our studies in the past. In fact, we have the biggest cohort of elite controllers in the world. And we have learned a lot from studying the blood, and the gut, and the lymph nodes, and the lumbar punctures, and everything we've done, and people in this audience. What we have learned is that to control the virus indefinitely in the absence of therapy, you probably need 3 things. You need to start off with a low amount of virus, which is key, and we think we can get there through various interventions. You need to deal with the fact that HIV, even during treatment, is a state of chronic inflammation, because as was shown in that second video, which was brilliant, it was actually a very complicated series of ideas that have been shown in these videos, but the second one in particular talked about how chronic inflammation leads to the exhaustion of the immune system, and the immune system basically stops working. If you stop the inflammation, the immune system works better. That, to us, I think, is a central part of our institute and central part of how we're going about this, is to reduce that chronic inflammation. But ultimately what you need are powerful killer cells, CD8-positive T cells, which are primed, they're supercharged, they're ready to go, they know where they need to target specific areas of the virus, and they live in certain areas of the body, particularly lymph nodes, where the virus is. We need all that stuff to happen at the same time, and if we achieve that, you stop therapy, the virus doesn't rebound, in theory. So, 3 things, right? Lower reservoir, better T cells, low inflammation. How are we going to do that in the clinic? Well, the first idea that we're going to work on in a study that's going to start off in the next month or so within the Institute in collaboration with our colleagues at Gilead, which is another critical part of our Institute, we're going to take, what we're going to do is we're going to take this concept that in people who become HIV infected, the vast majority of people, they have a high viral load, this peak here, viral load comes back down. They don't have much of a T cell. Their T cells are not recognizing the virus. They have very high viral load, okay? They go on treatment, they do okay. Then you have this other group on the other side, these elite controllers, people who have very powerful T cells, they're born with them, they control the virus on their own. What we're going to do in this first study within the Institute in the clinical trial is we're going to look at these people in the between, people whose immune system works okay, but not great. And we're going to look to see if we can actually put them on regular antiretroviral drugs and see if we can stimulate through TLR7 with a drug, which we think is safe, to actually take someone with medium T cell responses and make them more powerful. And we think if we do that and they stop therapy, the virus won't rebound. And we also think, very importantly, if we do that, even during treatment, the virus that's there will get killed and eliminated, and you have a smaller reservoir size. So, within the Institute and in collaboration with Gilead, right after the new year, and if you want to know why I'm a curmudgeon, all right, I've got to answer that question because I own it. It takes about a year, maybe even longer, to take an idea and move it to the clinic because of just the massive amount of paperwork, which can really basically, it's soul-sucking. It's turned me into the monster that I have become. I used to be a very sweet person. And you can ask anyone who works with me, you put a piece of paper in it and these get signed, and I start screaming and yelling. It's a bit scary. Anyway, so we've been working on this idea. We're trying to get this through the FDA, IRB, UC contracts, lawyers, so forth. We are all set. In about a month or two, we will be able to get up and going and do this study in the clinic. And what we're going to do is we're going to take GS9620, right? This drug that we've been sort of dancing around, right? It stimulates TLR7. It's a pill. Goes into the body, goes in the gut, goes in the liver, goes to your immune system, turns it on, okay? And turns it on subtly, so you'll get a lot of inflammation, and it turns on the cells so that they can actually kill HIV. We know it's safe, and we know it worked in the monkeys. And we're going to take people who are controlling virus on their own, who go on antiretroviral drugs, so people whose viral load is low but not undetectable go on therapy. They're going to get randomized to this drug or placebo, and then we're going to stop therapy. Okay? Now, this is a controversial thing in the field, but we're all becoming comfortable with this. Almost all these clinical studies that are going to look at cure in the modern era are probably going to have to require very careful, highly monitored interruptions of therapy. And we can talk about this in the discussion period if you guys want, how we think we can do this safely. And the reason why we need to do this is because, as basically everyone has said all day, we do not know how to measure the reservoir directly. The only way to sort of figure out what's going on is to stop therapy and to do it as carefully as possible. And again, I'd be happy to talk about how we're going to go about doing that. What we think is going to happen, certainly what we hope might happen, is that as a consequence of this intervention, HIV-specific T cells, which are present, will become much more effective, and you stop therapy, and the virus doesn't rebound. And we're going to basically start doing this in people who had a viral load below 10,000, but detectable before therapy, and we're going to start this hopefully after the holidays. So that's the first study. After that, in the summer, we have a second study that's going to look at not TLR7, but TLR9 to see if we can stimulate that. That's sort of a backup plan. And to be honest, I think that in some people, it may prove that this is all you need, but I suspect in the vast majority of people who have a lot of virus and so forth, we're going to need more than this. The other major component of the early curative studies that I think we're going to need to do require vaccines. So you give a vaccine, an HIV vaccine with these approaches, you combine them. That vaccine work will also be in the clinic here in San Francisco probably about a year later. But ultimately, this stuff is going to have to be combined. What about this issue of chronic inflammation? And again, it was highlighted in one of the videos much more eloquently than I can do. But what we have noticed and what many people have shown over and over again is that if you measure activation, T cell activation, inflammation, however you want to define it, in people with HIV who are untreated, it's pretty high. Okay? Half of CD8 T cells in the body can be turned on. And that's not good. That wears out the blood vessels, that wears out the brain, and so forth. And that's a major reason why going on therapy is really effective. Because when you go on treatment, as you go from that red to the blue, the amount of immune activation in the body plummets. It goes down very rapidly, dramatically, and almost, but not quite, becomes normal. And in particular in the tissues, we have found that it remains elevated. And we think that that elevated inflammation is having a subtle effect on T cells, making them exhausted. They don't work so well. We need to turn that off. This, by the way, is a concept which is revolutionizing cancer therapy, this concept that I'm talking about, this chronic inflammation, immune exhaustion, T cells don't work. Cancer immunotherapy, all these drugs that are now sort of changing the way we think about cancer, same problem. Cancers are a site of chronic inflammation. The T cells that are supposed to kill the cancer get into the inflammation, into these zones, they don't work. You block the inflammatory pathway through multiple different directions. The T cells work, cancer goes away, perhaps the HIV will go away. So this is a concept that is now really having a huge impact on cancer, kind of like antiretroviral drugs had on HIV back in the 90s. It's really dramatically changing the face of the field. And in fact, this whole concept of cancer chemotherapy is generally recognized as been one of the great advances in modern medicine. And I'm mentioning this because all of the pathways that have been identified in cancer which affect immunotherapy, people like Mike McCune and his group here in the room have been saying for years are having the same impact, same pathways in HIV disease. And what's convenient is that we can take the drugs and the knowledge that's being gained in cancer patients, where you can do more aggressive studies because the people are at much greater need than people with HIV, find out how these drugs work, what's safe, and then to turn it into the HIV cure world. And that's ongoing now outside the Institute. Multiple studies, I'm involved in about a dozen of them myself, either directly or indirectly, are trying to block the pathway to see if inflammation goes down. What the Institute is going to do, and is doing, is actually bringing our skills and our power to these studies because of our deep knowledge of immunology and virology, and we're actually leveraging the Institute funding to sort of get specimens from these studies, bring them to San Francisco, study them to see how these cancer drugs might affect HIV cure. And there's a whole series of these studies. Some of these pathways are quite complicated, but some of these studies are happening here in San Francisco. Some people in the room are actually involved in some of this stuff. And what we hope to do is to use the Institute expertise to figure out how basically to study the reservoir and identify novel ways to enhance the capacity of our lead compounds to actually work. All right, so one last slide, one last concept. Okay, I do not think that we're going to cure people with aspirin, or even with a less benign drug, just a single drug. It's going to require either gene therapy or a series of combination regimens given to someone who's on therapy for a limited period of time, but almost certainly going to be a combination approach. Studying combinations in people are complicated, right? You have to first do the studies by themselves, which we're doing now. We're launching a whole series of these studies. But ultimately, what I believe the Institute will do over the next 4 to 5 years is reaching out to our friends in pharma, reaching out to the other groups that are working with NIH funding, who are doing complementary pathways. Paul mentioned this in his introduction, to identify really the most viable combination, probably 2 or 3 different drugs given at the same time, which we think can actually have an impact in achieving our goal, which is a remission and cure. And I think, people often ask me when we're going to have a cure. I have no idea. But when we are going to have a regimen, a combination that's viable and testable, which is the goal of our group now, that actually could work, I'm hoping that we're going to have that by the time the current funding of the current cycle of the Institute is over in the next few years. So that's an optimistic perspective. I started this job back in 1993, and what I saw during the first 3 years of my career was what everyone else in this room is well aware of. We went from a fatal disease to a disease that was controllable. No one in 1993 had any idea that we would actually be able to control the virus, but yet 3 years later we did. So I'm sort of optimistic that we will make great success, and that kind of where we are in this science is kind of where we were back in the late 80s, early 90s. We're beginning to have an envision what a combination might look like and started working in that direction. So, Kevin, so I'm going to turn it back to you, and then thank you very much.
Stay here, because we're going to go ahead and we'll sit down. So let me invite all of the speakers now to join us here on the stage. We're also going to be joined by Matt Sharp, who many of you know, longtime AIDS activist here in the San Francisco area, done a lot of work over the years, but also particularly in Cure. So he's going to join us for the conversation. Yeah, I see people raising cards, so some of the team are going to come around and get those cards from you if you have questions. I've already started to work through some of the cards. We have some really great questions here. So we'll just continue with the conversation once we all get up here.
Oh, this is in the center.
Anywhere's fine, guys. So they've been paying attention because we have some great questions in here. You know what? We have exactly about 37 minutes left, but at any time, feel free to get. I mean, we're not going to be offended if you get up and go to the bathroom or
I'll come with it.
Or grab some coffee, which is in the back of the room. Feel free. There's no problem at all with that. All right. Let's get going because there are a lot of questions here about the reservoir, and I'm not entirely sure who wants to handle each of them. But let me begin with this, which is kind of a broad question. Are there other examples in nature of latency like we see in HIV?
Let me try to start.
Paul.
Because I'm an oncologist by training, and really the concept of a latent reservoir in HIV is not unlike the concept of a latent reservoir of cancer cells in somebody who's had a primary treatment. So there are some cancers, malignant melanoma is the best example maybe, where depending on what the initial cancer looked like when it was taken out, you know it's going to come back. And it can come back 5, 10, 30 years later, but you know it's going to come back. And yet we can't find those cells in the body. And as Steve was saying, some of the same mechanisms might be very operative in terms of keeping those cancer cells under control and in preventing the immune system from keeping them under control as exhaustion happens. So I think to me, that's a great model.
What about with viruses? Are there any other viruses in nature that behave this way?
Well, I mean, herpes viruses are renowned for going latent in neurons and then coming out and causing lesions. HIV is unique. And, in general, viruses pursue one of two strategies to persist in their hosts. They either chronically replicate, HIV does that, or they form latency. Most viruses choose one or the other. HIV does both, which is unique.
So it kind of stands alone in that regard. So the formation of the reservoirs is a question that people are trying to understand. And one of the questions asked here is, what about people who start antiretrovirals late in the course of their disease or who are diagnosed late and therefore have started antiretrovirals late? What do we know about their reservoirs? Are they larger? Are they smaller? Are they harder to get at, easier to get at? What do we know about people who start antiretrovirals very late?
Yeah, so people who start late definitely have larger reservoirs, and not just that, but the immune system works less well the later you start HIV meds in the course of the infection. So all the strategies we're trying to harness to kill infected cells with therapeutic vaccines, TLR agonists, stimulators, and the like, it's possible they may work less well if people have started very late. And so you look at people who start very, very early in the course of their infection, the first couple of months, they clearly have much smaller reservoirs and they have much more preservation of the immune system's function, which may make it easier to eventually clear the reservoir, we think.
So put that into perspective then. How soon after a person is infected do you start to see the reservoir forming?
Yeah, so Steve, do you want to?
So there's we're working on that very carefully now because we have people coming into the cohort now, the options cohort, who probably been infected for 10 or 15 days, and we and actually a group in the military are doing the same thing. And what they're seeing is that from about day 15 of the infection to about day 28, for the week 2 to 4, the reservoir gets established and goes up by 100 to 1,000 fold. A lot of that happens in that critical 2-week window. And that is a very challenging time for people to start therapy. They just found out they're infected. They have other things on their mind. They don't want to jump on these combination regimens. But it's actually almost every minute counts during that period. After that happens, the reservoir is more or less established. And then the goal of early therapy then is actually to preserve the immune system, as we've been discussing. Because that you can, the earlier you start, even after a year or two into your infection, you're actually still preserving a fair amount of immune function. But the reservoir gets established pretty quickly.
That's not so different in terms of time frame as to the antibody response, is it? So you're seeing the seeding of the reservoirs and the emerging antibodies roughly in that same sort of time frame, very, very early in infection.
And the assumption is that the antibody response in the T cells is what stops the reservoir from expanding beyond that critical period. So once the virus takes off at around day 14 of the infection to around day 28, it's a race, right? The virus is going crazy. The immune system is just getting revved up. The virus typically wins. By the time the immune system gets up, the reservoir is too big to do much of. The exception being sort of these elite controllers.
Right.
And people who start therapy very early.
So I just want to mention, if Weinberger's right about the planned latency, it could be established as early during the eclipse phase. Even before we can detect the viral infection is occurring, there could already be latently infected cells being established.
Yeah, and one other point to add, another critical component of persistence is that the virus can colonize practically every tissue in the body, the spleen, the lungs, the brain, and there have been studies where, at least even in animal models, they've been able to demonstrate that, within the first week you can find HIV in multiple tissue sites, like it's already crossed the blood-brain barrier and established, a persistent infection within the brain, which is kind of an immune-privileged site. It happens very quickly. The other point I wanted to add, I showed umpteen different tools that we're using to measure the reservoir. Every single one of those measurements that we've attempted when we've compared early and chronically treated individuals, except for one measurement, all of those measurements were smaller in individuals that started treatment within the first few months of infection. And the point that Steve made, the critical point is a lot of it probably is not just a matter of the initial establishment of the reservoir, but it really is the preservation of key immune responses that accelerate the decay of the reservoir after it's established.
Well, you offer a nice segue into the next question, because in your talk you mentioned that tissue is probably where the money is, right? That what we're measuring in the blood may not be very indicative of what we need to know in that tissue, but you also think tissue is very difficult. In this context. So the next person is asking, what are the best animal models that we have that might inform us if we're going to be talking about tissue?
Yeah, so the choices are very slim. I think probably most people might be aware that there are many humanized mouse models that are coming on the scene, but the way those work is essentially they have a component of human immune tissue that's transplanted into those organisms, so it doesn't really allow you to study the tissue radiation of the virus in kind of a physiologically normal manner. So the closest thing are the non-human primate models, I would say. But even then, I think there's a lot of value that comes out of those models, but the host itself and even the viruses that we have to use to infect those animal hosts are different, slightly different, but they're different in some ways that I think are important that might change the situation. Any guys have anything to add on that?
Yeah, I would add one point, and that is, while the humanized mice are not a perfect model, they don't form a perfect lymphoid system. It is possible to modify the hemopoietic stem cells that are giving rise. Alex Marson, who's here today, along with Jennifer Doudna, have pioneered techniques for genetically editing using CRISPR technology to edit the genome and knock out or knock in genes. And so now those hemopoietic stem cells can be put in to the humanized mice, and the effects of those genes on the latent reservoir can be studied. It could be the combination of these incredible genome editing technologies and humanized mice offers a unique opportunity in latency.
And I'd just add, too, that while the primate model is good and with limitations, but it's also really expensive. It takes a long time as well. So if there could be more use of the smaller, the mouse, for example, that research can go much faster. And so I think there's a real pressure to find a better model.
All right, so we dove right in here, but let's take a second and open the lens. Because, Matt, you've been sitting here and you've heard all the presentations, and our scientists have been talking. How are they doing? What's your sense about Cure Research?
Ruh-roh.
Well, first of all, thank you for inviting me today. It's always a pleasure and honor to be invited on any of these panels here, especially at ECSF and Gladstone Institute. You know, I'm sitting in the audience. I've been a little bit separated from some of the work that's been going on in the last year, and it's been really great to get this update today. Although I have to say that even as an activist that's been following Cure Research for this amount of time, it's a lot of it's way over my head still. But I will say it's really encouraging to see this project go into the weeds, like really into the weeds, and determining the measurements, you know, the level of intensity and looking further and beyond what we already assumed we knew, and then also to the level of getting into the clinic, I think is really critical, especially with the shock and kill approach. But also with, you know, I'm encouraged with the other approaches that, thank you for mentioning the gene therapy approaches, which, you know, many people are entering into those clinical trials as we speak. A lot of them with the Fred Hutch Institute up in Seattle. So that's really encouraging to see. So I think, you know, what we're getting to at this level is an understanding of deeply into the science, which is the basic science, which is what we need, obviously, to understand how we move further into the clinic, which is also happening. So it's really encouraging to see. It's also a little bit daunting for me as a patient, knowing that I probably won't be cured. Maybe, you know, we'll keep our fingers crossed. But to see that, you know, the legacy of what's happening at this point will one day get us there. I really.
Is it fair to describe you as encouraged, optimistic?
Oh yeah. I'm still very encouraged and optimistic, and I.
Despite what Steve says, I didn't.
No. Actually, in addition to what he said, I mean, he seems to be always. He always ends with that optimistic.
He is underneath that
But
Underneath
you need to understand. So Matt is a classic. Can I tell your story?
Yeah.
So Matt went into one of the original gene therapy studies with Sangamo back, I don't know, 4 years ago?
No, it's been 5, 6 years.
5, 6 years ago.
Yeah.
And we had we had a theory what might happen, but it was completely made up. We didn't really know what was going to happen. And we took his cells out of his blood and we gene modified them and we poured it back in and interesting things happened. We were surprised.
I was on therapy.
He's always staying on therapy. So, but we took his T cells, we gene modified them, we put them back in, and the amount of virus in his body went down, but his immune system got much better. Right? Your T cells went up, and his immune system started working better, even though we didn't really do anything specifically for the immune system. So we brought him back in, and you're actually, I think, one of the first people who we encouraged or twisted your arm to give us a lymph node.
Yep.
Yep. So we practiced on him. It didn't kill him. We took one of his lymph nodes and we sent it to a bunch of scientists, and then they came up with these data suggesting that all you have to do is protect a small proportion of the CD4 cells if they're in the right place, the lymph nodes, and you can do great things. And so it's a classic example of what we do very well here in San Francisco. We do these early studies, make some observations, generate some riddles, do some puzzles, get some tissue, and the field moves forward.
Well, can I just add one more thing to that? And thanks for bringing that up, because I was hoping somehow we would discuss it today. is the sort of what I call the incremental sort of advantages to these trials that we're seeing. And one thing we don't really understand is that we feel like with people like me in this first Sangamo trial, we're seeing basically a resetting of our immune systems. My T cells were doubled in that trial, as most of the participants were, and we're still looking into the reasons behind that. Possibly maybe a new cell that's created by just simply doing this approach, this gene therapy approach, which helps stimulate the immune system. I hope I'm explaining that correctly. But anyway, I'm still, after 5 years, my T cells are still at that same level. So.
Are you able to participate in other gene therapy trials?
I was thinking about that when I was sitting down in that chair waiting to get up here. It's unclear. It depends on the trial, I would assume. Yeah.
So, all right, let's move on, because there's a number of questions here where people are asking about gender differences in cure research broadly, but specifically gender differences in reservoirs. Do we know anything about gender differences in reservoirs? Do we know anything about how hormones may affect the reservoirs? What do we know? Anyone can
Before we
speak to
get
that?
to the science, let me just say that Rowena Johnson, who couldn't be here today from amfAR, is on our group, and she's very interested in this, and that's why one of the reasons why this amylase treatment interruption trial will have, we hope, 10 out of 30 people participating will be women. So.
Well, I was going to lead to that in the next question, because someone is also asking about how important is diversity in the context of the studies that we are doing, and what are we doing to ensure that diversity? Maybe we can build on that, but come back to the science. What do we know?
Yeah, so we've known for a while that the biology of HIV in men and women, there are differences. So in the absence of therapy, women tend to have lower viral loads than men at any given CD4 count. Others several years ago have demonstrated that the response of the innate immune system to the virus is enhanced in women compared to men. So there are immunologic differences that are likely related to hormones. And then most recently, funded by amfAR, we were able to recruit a large cohort of HIV-infected women, and working with John Carn in Cleveland, his group was able to show that some of the female sex hormones, like estrogens, actually suppress HIV reactivation, promote latency. So it may keep HIV, in its hiding place, allow it to persist better, and so that's led to the theory that perhaps anti-estrogens may help release HIV from its hiding places, and so there likely are important gender differences, and perhaps even gender-based therapies that may come out of that.
So that automatically, I think, answers the question that gender is important in the context as we move to clinical studies. Steve, what are we doing to ensure that diversity in our trials?
Well, this someone just called me about an hour ago and yelled at me because apparently I was in another meeting. I said something about it's hard to study women because they have small veins, and then they
Did you say that?
I did. And then I promised I wouldn't say it again, and here I am an hour later saying on the stage. Women and men are different, and there are a lot of challenges with women in HIV that are unique. And to be honest, Kevin, no one has ever done what amfAR did. You gave us a big chunk of money. I'm not sure you knew that, to go out and build a cohort of women, and it was lots of challenges in doing that are different than building a cohort of men. And I think that if the field is very serious about trying to figure out whether therapeutic interventions are different in men and women, we need to do studies just to answer that question, which are challenging, which are studies just of women. But having studies of 10 or 20 people, who 2 or 3 are women, aren't going to advance the field. I probably get in trouble for saying that. Fortunately, this is not being videotaped. But I think that if you want to answer this question, we should, based on this work that Peter just mentioned, we need to do this in a very specific and highly resourced manner.
So I forgot to mention that we are videotaping. Seriously, we are videotaping, and I also forgot to mention that we're broadcasting live through Facebook Live. So the 2 video cameras in the back of the room are actually set up, and we're Facebook Live. So thank you and welcome to all of you who are online and watching. I understand we're just over the 3 million mark, Steve. No, I'm kidding. But we are videotaping as well, and a video, in case you want to go back and watch any of these presentations, will be available
There's some
afterwards.
tweeting going on, too.
Yeah, we got tweeting and Instagramming the whole bit. Yeah.
So thanks for that little bit of levity here. I think we needed that. But what I wanted to add to that is the question of diversity in terms of race.
Yeah.
And that's all often forgotten. I'm really thrilled to see that women are going to be included in this study, this first study from the get-go, because I think that's what we forget is that we need to plan that in the beginning phase and not look at it afterwards and say, oh, oops, we forgot women or we forgot minorities. So, and that's a good point to really design a trial starting from the get-go with women only or diverse populations only. So.
That's great.
There is a literature, Rowena is reminding me of this, in terms of TLR7 agonists. Women, dendritic cells from women respond less well to TLR7 agonists. And of course, that's a centerpiece of our toll-like receptor stimulating system. And whereas TLR9 agonists are equal between men and women. So we have to pay very close attention to this gender difference.
Back into the weeds for a quick second here. Somebody's asking about the reservoir. You've talked a lot about T cells. Can latently infected cells exist in other cell lines?
In other cell lineages in
Yes.
vivo.
Macrophages?
That's a $1 million question. So I think people have always been really, really interested in the idea that myeloid cells like macrophages can serve as viable reservoirs, and the question's been asked whether they contribute meaningfully to the size of the reservoir. I'd say there's been decades of very intense research to figure out if macrophages are legitimately contribute to HIV persistence. The big challenge with macrophages is what macrophages do for a living is they gobble up other cells, right? So if you find a piece of HIV DNA inside a macrophage, it's very hard to really unequivocally demonstrate that virus actually infected that cell. It actually might be present in that cell because that macrophage went and gobbled up a T cell nucleus that happened to have it. So I think it's a very difficult thing to prove unequivocally, but many people have tried. I don't know if you guys drank the Kool-Aid one way or another on the macrophage contribution. Yeah, I'm kind of agnostic to it.
So we don't know. We're not sure.
My opinion is we don't really know.
Okay.
You can certainly in the laboratory you can force your way into a macrophage. Like you can get virus to get into a macrophage. Whether it happens appreciably enough to care about in an infected individual, I think remains to be addressed.
Okay. Next question. Won't latently infected cells eventually die if a person is on ART after many years of having undetectable levels of virus in their blood? How long would it take for these latently infected cells to eventually die?
Decades.
72 years is the
72
current.
years. The Silicano.
I didn't make that up. It's actually 2 studies that more or less 70, 75 years.
So the answer is we can't wait it out.
Well, you can if you're 5 years old when you get HIV. But
Yeah. Or if aging research proceeds their lifespan becomes 200 years, then maybe it becomes
Then it might
But
make sense.
I actually would even challenge those numbers, that I think that there is a component of homeostatic proliferation in which the latent reservoirs, under the influence of cytokines, they divide and they maintain. So I think there's another school that would say, no, you're never going to wait this out. It's going to be there forever unless we attack it more effectively.
All right. So we've had a couple of questions along the lines of analytic treatment interruptions. A couple of people remember the treatment interruption studies of an earlier time. And some people obviously don't have very fond memories of those studies. They feel that they were risky. And they're asking the question, how much risk are we assuming in analytic treatment interruption studies today? And you've described one, Steve. How does your IRB respond when you go and say we want to do an ATI study?
the FDA and the IRBs, the ethics committees, have been extremely understanding, and supportive when we explain what we're doing, okay? This is really different. So back to studies that you mentioned, Kevin, where we take people, stop therapy, and tell them to come back every 3 months until your T cells are below 200. And by surprise, right, they got more likely to AIDS. So those studies were not well designed, and I don't think a lot of people even supported them back in the time they were being done, including myself. We're not going to stop therapy and tell people to come back in 3 months. In fact, we're going to actually probably, if anything, go a little bit on the extreme. What we do in these interruption studies now is we stop therapy, and the virus typically doesn't come out until at least week 2. So we see people at week 1 and week 2, and then we see people 2 to 3 times a week when the period of time is most likely the virus can come out. And we send nurses out to homes and so forth, so we don't try to overburden people, but we monitor the virus very carefully. And once we see the virus, and we just had a situation like this last week, a person whose virus was like 50 copies. We bring them in immediately, we start them back on therapy.
Even at 50 copies?
It's all the protocols are different, but we were pretty sure that his virus was about to take off, and we don't need that, right? We don't need to watch the virus go crazy. We just need to know when it comes out to answer the question scientifically. So if people are willing to be monitored very carefully, we can get people on therapy when they only have viremia for a few days. And to be honest, the major risk that I worry about is not to the participant, because nothing really happens to the immune system in 2 or 3 days or 4 or 5 days. It's a 10-year disease. What the main risk then is actually sexual partners, because you don't know when this is happening. And so you have to be careful during this period of time. So those are the types of interruptions that we're doing now. Fundamentally different, highly monitored, and we think we can get people on therapy without any major
population of people too. It's
a whole different question being asked than before.
I remember doing a treatment interruption study, that acute study in the ACTG, and we didn't know anything. We didn't really understand that, that there's a relationship between viral load and transmission. We did this study, and we didn't even ask the question of transmission to partners. So we've learned a lot, at least since then.
Why is stopping treatment so important to the study of cure research? People are asking, a couple of people have asked this question, what makes the ATI so important to our understanding of cure research?
Because the super geeks here on the panel, Satish, have not figured out how to measure this stuff, right? He's got all that fancy science, but he didn't actually say at the end of the day, it's all nonsense. None of this stuff actually tells us anything useful.
That's the whole point. That's why I'm saying we need to do a treatment interruption so we could evaluate all these tools to see if they tell us anything. And I can't think of any other way, unless if you had a magic 8-ball. There's just no way.
Break that down, though. Help people understand what you're saying, because you've got all these fancy tools and fancy names, but what is it that stopping treatment tells you?
Okay, so let's just, I guess the way I like to think about it is in the real world. Like, let's say Steve, I wouldn't gamble on it. No, I would gamble on it, actually. But let's say Steve comes up with some successful eradication strategy that we want to try in the clinic. He tries it. So ultimately, the goal of these eradication therapies is to be able to take people off of treatment indefinitely. But ultimately, we need to have some tool that tells us whether or not the thing that Steve tried in the clinic is likely to result in lifelong remission, or at least an extended period of remission without antiretroviral therapy. The only way we can develop that tool that says, yes, I did some little assay in a tube, something lit up green, that means that it's safe to stop antiretroviral therapy in this individual. The only way that we could come up with that little tool that glows green when it's safe to stop therapy is if we experimentally stop therapy in large numbers of individuals, measure these things when we stop therapy, and determine if any of these things actually allow us to predict that. I personally cannot think of a way that we can come up with a way to measure these things in the field reliably, or know that we're measuring them reliably without stopping therapy and evaluating the tools.
The only way we knew the Boston patients weren't cured is by stopping therapy. The Mississippi baby, the same thing. It is the gold standard for figuring out whether or not your intervention was effective or not. There's no replacement for it.
Yep.
And it was why we reacted so skeptically to this news story a couple months ago, I guess, about an English study where somebody was said to be potentially cured, but it turned out that he was still on antiretroviral therapy.
Right.
You can't.
What do you make of that?
Yeah.
Not much.
And can I just say that we've gotten to a level where
we understand the safety involved here in doing that. And we've spoken at length with the FDA. We've had forums with the Collaborative for HIV Research. We brought in professionals and experts and community to really hone down what it's going to take to do these as safely as possible. So I think, we can't do it we can't do these trials and go forward and learn anything unless we're able to do that. And I think, if we do it with the fine-tooth comb, case by case, which is exactly what the FDA has said, case by case, then I think we can do it safely and learn something.
Yeah, and I think it's important to keep in mind that the ultimate goal is long-term risk reduction, really, right? That, by doing these carefully executed, experiments where we can, really ensure safety as much as possible, that in the future, we will not stop antiretroviral therapy unless if we have a really, really good indication that there's going to be lasting remission and benefit when we actually do.
So universal agreement on that point? Necessary, the gold standard. I'm being signaled that we have about 6 or 7 minutes left, and you all know what that means. We're going to enter the lightning round. We've gotten a number of questions which are not necessarily on point of Cure or Cure Research, but nonetheless I think are important questions that have to do with sort of the world we live in today, which is probably, for most of us, a different world than it was just a month ago. So a number of people are asking questions, and I'm just going to throw them out there, and you guys can pick and choose who wants to answer them. What's the best way to learn about Cure trials?
Your new videos.
Well, that'll tell you about the Cure strategy, but how does somebody learn what trials are going on in Cure Research? Where do they go? They can't all call you.
I know that the Treatment Action Group maintains a pretty up-to-date, so TAG.
Well, they're all registered in NIH
clinicaltrials.gov.
Clinicaltrials.gov.
Not the greatest interface, but
But an email to me might be the best way if you're local.
Okay.
Because we know about most of the stuff.
Okay. A number of people have expressed concern that in a new Trump administration, funding for research and funding for AIDS in specific is likely to decrease. How do we advise our local, state, and federal reps to help us in this fight? Anyone?
Well, we have to remind people that the epidemic's not over. I think some of the concern that we have about Cure Research is that people might mistakenly think it's already done. So it's not. We're just starting this. And this epidemic is coming under some better control, but if we turn our back, it will explode again. So I think reminding people about what it used to be like, because we don't want to go back there. But I think we're all concerned about the state of the politics.
Activism by groups like ACT UP was critical in getting funding and changing the medical establishment. Does this kind of activism have a role in 2016 and going forward? Matt, you have any thoughts on that?
Well, tied to the last question, yes. I mean, I think in terms of what's happening with our new president-elect, which I'm even reluctant to even say that word. I'm so angry about the whole thing that I think we're going to have to resort to some of that in regards to what some of his decisions, really, and even around the cure arena specifically. Why not? I mean, if it's going to take what it's going to take.
Well, and I think we should not be complacent. People say, well, both Republicans and Democrats support Ryan White and support PEPFAR, and I just don't think we know that anymore.
I'm not sure we do either. I think a lot of the folks that were in Congress, both Democrats and Republicans, who were there for the creation of PEPFAR are no longer there. And I'm not sure it remains a bipartisan issue anymore. And I do think that, in the changing world that we face, in the changing environment of healthcare, medical research, all those things, there's a difficult road ahead. I think there's no question about that. So just come back to the cure for one more second here, because someone's asking, let's assume for a second that we do find a pathway to a cure. What are your predictions about our ability to deploy it around the world? 37 million people, 38 million people living with HIV. You said, Steve, it's going to be combination therapy or it's going to be gene therapy. What are the implications for that, for deployment?
Well, we did a study with the Gates Foundation, which asked the question: what does a cure have to look like for it to have a global impact? Okay, and it has to be safe as treatment. It has to be effective. Either get rid of get virus completely, or keep it so low that you don't have any transmission risk. And it's got to be scalable. And they basically did a lot of mathematical modeling what it had to look like. It has to be something that could be administered in a small clinic for a short period of time, say up to 6 months, and can't cost more than $1,500. So that is what it should. And the approaches that we are talking about today, TLR7 agonist, which is a pill, which is not that expensive to make. You take it 10 times over a period of 6 months, no problem, no side effect. You add on top of that a vaccine. Vaccines are safe. Some of these anti-inflammatory drugs. So in my mind, I can imagine that there is a finite 6-month regimen, 2 or 3 different drugs that you can add on to a stable regimen that will be safe, don't require monitoring in a hospital bed. That could, in theory, work. For me, that's a game changer. That would have a global impact. The gene therapy work, if you want to cure someone with HIV and you want to do it as quickly as possible, gene therapy is the way to go. We have the Berlin patient. We have the successes that Matt has been involved with. But those are not going to be game changers globally. I don't see that having a global impact. Some people do.
Well, maybe not in the short term, right?
Not in the short term,
if you think about the very history of medicine itself,
Fair
think
enough.
about things
I
like
agree.
dialysis or laparoscopic surgery or any of those things. The very history of medicine is that we take things that are difficult and complicated, dangerous and expensive, and we make them simpler and easier and cheaper to deploy. And if HIV follows that same path, the road is a long one, I grant you, but eventually we will find ourselves to a place where we can.
I would say that if you're talking cell therapy as opposed to gene therapy, if you have a therapy that requires preconditioning with chemotherapy, I do think that's not going to be scalable.
But can I give an example of how this is moving forward, and what you're referencing is the technology at Fred Hutch. They've created what's called gene therapy in a box. And it's this young investigator who's created the technology around the apheresis procedure and all of that technology. Instead of moving into a completely other clinic to do that work, she's created it into a simple box technology. It's amazing. Look it up online. And that's an example. And that would be expensive, but I mean, it's still...
I would offer a sobering point of view, and that is, even with antiretroviral therapy and spending billions and billions of dollars, we are still not reaching half of the individuals in need in a developing country. What will drive a cure in the developing world is cost, because governments are recognizing we cannot sustain the treatment of 37 million people for the rest of their lives. It is far cheaper to come up with a cure than to that. And so with precious global health dollars, I think the fact that you could get people off of antiretroviral drugs, remove that burden, that billion dollar
Stop transmission.
Stop transmission. Yep.
Absolutely. That's what's going to drive this in the end.
One other point I want to make on that, and Rowena actually made this point in a perspective piece or a review piece that she wrote, I think, in AIDS Research and Human Retroviruses, is that almost the entire cure agenda, all of us are always talking about adding things to antiretroviral regimens. That's kind of the overall strategy for obvious reasons. We really want to shut off replication before we do something in there to destroy infected cells. But ultimately, it would be great to have some parallel investigation into approaches that don't even involve having antiretroviral therapy on board, which seems kind of even crazier, but probably not impossible. And that would be a huge game changer, is if you could try something in the absence of concomitant multi-drug therapy.
Yeah. I said this a year ago when I was here in San Francisco, but I think it bears repeating. If you need evidence of why we chose UCSF for the amfAR Institute for HIV Cure Research, I offer to you the 6 gentlemen sitting on the stage today. I'd like to offer their thanks as well. Just outside and on the balcony as well, we have a reception set up. Please feel free to hang around and ask questions. There's alcohol, which means Satish will be staying for a while. Please come on out and have a drink with us.
Where am I? But you're right.